Good morning, everybody. Welcome to Cincinnati, Ohio for the Catalyst Proposers Day. We are really excited to have you all here. Really appreciate you being here, especially in the quick turnaround style that we have at ARPA-H. And so thank you for your attendance. We also have over 350 people register virtually as well. And so this is a hybrid meeting. And so we'll have mics and things that we pass around for Q&A. I'm Andy Kilianski. I'm the program manager here at ARPA-H that's going to be running the Catalyst program. If the ARPA-H people in the room could stand up, I think most of them are in the back there. Those are my teammates. If you have any questions throughout the day, anything for us, please track them down and they'd be happy to answer. For the in-person presenters and the virtual presenters, we'll have about 1 minute of Q&A with the exception of our contracting session. And so we'll have a microphone that gets passed around and we'll be able to answer a few questions here in person. If we don't get to them during the run of show today, we have a running Q&A page that's going to capture all of the questions and answers from today's session, as well as any general Q&As from the one-on-one sessions this afternoon. So again, thank you all for being here in Cincinnati. We really appreciate it. We're going to run through first really what Catalyst is. I'm going to give a little bit of an ARPA-H overview as well, but really provide an opportunity to highlight some of the things that we put into the Catalyst program that I'd like you all to focus on. We'll then hear from Dominic, who's our business and financial manager on the team, with a little bit of the details about the submission process and the dates. Then we're going to hear, we're actually very lucky to have two of our contracting leaders from our business innovation division at ARPA-H. They're going to talk about the contracting strategy that we're going to use for Catalyst. It's a little bit unique, and so we provided some time for them to discuss that strategy as well as answer any questions. And then we're very fortunate to have three government partners who are going to discuss their perspectives on the Catalyst program from the FDA and the NIH. So not only to discuss how we're going to be working together, but also additional opportunities and areas to work with the U.S. government across HHS. Okay, so let's kind of dive right in if we can. Next slide, please. So I'd like to spend just a few minutes introducing ARPA-H for everyone. Hopefully you've heard of us, and certainly if you found your way here to the Catalyst Proposers Day, you've got some awareness of what ARPA-H is, but I just wanted to run through really quickly a little bit of how we operate and how we're configured. So our mission is a simple but extremely complex one. Our job is to fund high-risk, high-reward research and development to accelerate better health outcomes for everyone. Right, so our goal is to really be pursuing research that's going to push the boundaries of what's possible, but also be translational, because at the end of all of our projects and programs, we want to have patient impact. If you're familiar with ARPA-H, you're probably familiar with DARPA, the Defense Advanced Research Projects Agency. And so the ARPA-H model is modeled after DARPA and the successes that DARPA has had in innovating in technology with relevance to the military and national security, and the way that those discoveries and innovations have translated into impacts that we all leverage today. So we are a brand new agency within the Department of Health and Human Services. So we've been around for a little over 2 years now. We're an independent component of HHS. Our job is primarily extramural funding. So we don't have laboratories. We have a very lean management structure. We're capped at 210 government employees. So we're small. But that allows for us, again, to stay nimble and to really focus on the external parts of the funding that we want to provide. We are not a grant-making institute or institution, so we primarily do contract-based types of research. And so milestones, deliverables, assessments, it's a very active style of research that we fund at ARPA-H. And we also are fortunate to have a unique relationship with the FDA that allows for them to come into our programs and understand what we're doing, understand the technology that's in development, and really be a partner in the development of those. We've been appropriated money by Congress in which we're spending, and our agency is unique in that all the leadership and program managers have a 3 to 6 year term. And so there's a lot of turnover, and that's by design. We want the best and brightest ideas coming into the agency, and having that turn and that churn allows for us to continue to refresh the ideas and concepts that are coming into our page. So our ecosystem is pretty much everybody, right? If our goal is to impact the health of patients and health of Americans and everyone around the world, then that's really going to include everyone to be in our ecosystem. So we have a lot of interagency partners that we work with across the government. You're going to hear from some of them today. We work very closely with patient advocacy groups and patient populations and understanding really the impacts that we want to deliver to Americans in the healthcare ecosystem. And then for us at ARPA-H, we're judged by translation. And so it's great if our performer teams publish papers and really contribute to the state of the science, but we also want those things that are going to be impactful to people. So you'll see that throughout the Catalyst discussions today. We're looking for that product. We're looking for that capability, those things that, again, you are all going to be working on to develop in support of the objectives of ARPA-H. And so that means industry partnerships, really important. That means interactions with the investment community are really important. ARPA-H, we see ourselves as a non-dilutive investor in between something foundational that the NSF and NIH is funding and really what a venture capital firm might invest in or what might become a strategic partnership or an M&A. We want to be that de-risking partner really across that valley. So I kind of already covered this. Our programs are typically 2 to 4 to 5 years in length. Catalyst is 36 months in a first phase with an 18 months in a phase 2, and we'll talk about that. But the way that we construct this is that we build these programs to really last the test of time and program managers. And so it's very possible at the end of the Catalyst program, I'm not going to be at ARPA-H. Right. And so we're configuring these efforts and really bringing in teams that are going to be specific to the objectives of the program, right, and not any one program manager. So at ARPA-H, we've got really 5 key mission focus areas, and these have become our mission offices within the agency. So Catalyst and myself, we fall under the Health Science Futures mission office. I'll talk about that a little bit more in a slide. And then we have 4 other mission offices: Scalable Solutions, Proactive Health, Resilient Systems, and Project Accelerator Transition and Innovation Office. And so these offices really span the access and care continuum, everything from really transforming how we do biomedical research and development, like in Health Science Futures, to scalable solutions of getting what we make out there to everyone in a way that's equitable and accessible. And so each of these mission offices has a solicitation. They have an open solicitation, and so I would encourage everyone to go take a look at those as well. And then the programs that we launched at ARPA-H fit within one of these mission offices. So HSF, thinking about the Catalyst, kind of like what is our goal within the program? We are really focused in HSF on tool development, platform development, things that are disease agnostic and that are applicable to a wide variety of outcomes. And so the way that we think about funding research in this space really has sort of these 4 characteristics. And so adaptable approaches, right? As we go through the course of an ARPA-H program, it's likely that the science is going to change. Our objectives might change, our outcomes might change. And so really having flexibility in the way that we configure our work together is very important. Obviously, we're looking for innovative methods and we're looking to expand what's currently possible in the solution space. And so think of those areas of white space where, nothing exists today, like the digital twin for human physiology for ADME-Tox. Those are areas in which we want to fund and push technology into. And then finally, to get these things done, very likely need to be interdisciplinary, multidisciplinary teams. And so people who really span a lot of different expertise areas who we can pull in to a certain program or a project to help accelerate innovation in that area. Okay, so with that, I'm going to dive right into Catalyst. And so we can go ahead to the next start. So Catalyst, as you're likely getting familiar, we like acronyms and usually very contrived acronyms in the ARPA world. And so Catalyst stands for Computational ADME-Tox Analysis for Safer Therapeutics. And so the goal of the Catalyst program is really to transform how we get to first-in-human approval. Right. So right now, the process is a linear process. It takes a lot of time. It's very expensive. Right. And there's a very big bottleneck at those IND-enabling safety regulated studies that really prevent a lot of creativity and access into those pipelines from across the community. If you're an academic lab and you got a really cool potential candidate, if you're a small company or an NGO who's working on a rare disease or a drug that has a small market, paying for access to GLP laboratory capacity for higher order mammal types of animal models like non-human primates, and getting in the queue to do those studies, which sometimes takes years, can be really prohibitive, right, to the innovation that we want to be driving at ARPA-H. And so the goals of Catalyst are to transform how we do those regulated studies currently using in vivo models today and really build a physiology-based predictive set of tools that we can use for ADME-Tox, PK/PD, and that are going to allow us to do other things in the future. And so while the goals of Catalyst are very well laid out in our solicitation, we see a future of digital clinical trials, in silico phase 0, phase 1 studies. That's the world we'd like to enable with the Catalyst program. So we already use a lot of computational tools, right, in drug development. A lot of what we're using is upstream, right, that can help us de-risk compounds that are in the discovery phase. And we're seeing companies more and more coming up with really cool AI-enabled types of tools that allow for the pharmaceutical sector to, again, de-risk the upstream process. And so how do we better understand pathways that relate to a disease? How do we better understand protein targets that we might want to drug? How do we go from, a billion chemical moieties down to 10 to start our wet lab work? What we'd like to do in Catalyst is bring those innovations and really accelerate the ability to use these in silico tools for safety evaluation. Right. So our goal in Catalyst is to come up with tools that fit that middle space. And so how do we better predict toxicity and safety? How do we better determine dose range for clinical studies? How do we look more closely at drug pharmacology, metabolism? And then how, through what we are building in the Catalyst program, can we inform trial size, population recruitment, and doing better phase 1, phase 2, and phase 3 studies? And really, how do we get to a place where we're going to regulators with an IND submission that is more predictive of human outcomes? Right. The reason why this is such a big deal, and I probably don't have to explain much to this audience, it takes 10 years and $2 billion to get a drug licensed, right? And that's if you're one of the lucky less than 10% of candidates that gets all the way to the end of that process, right? So the current toolkit has an over 90% failure rate, and those failures are extremely expensive, especially when they come later in clinical development. And occasionally we get safety and efficacy signals post-license, which is really suboptimal. And so what we'd like to do with Catalyst is determine ways to parallel these activities, right? So that you're doing your AI-enabled drug discovery, you're also in parallel doing your AI-enabled digital clinical trial, right, with simulated humans. We want to get to a place where instead of years of preclinical development and discovery and $500 million, it takes weeks to months. And we can do this in a way in which those capacity issues that we have in IND-enabling studies are circumvented and made more accurate by the in silico tools that we want to develop as part of Catalyst. Right, so cost is a big driver here. Time is a big factor. And then, especially at ARPA-H, we have large equity and accessibility metrics. And so there are many drugs that are potentially life-changing for patients, maybe smaller patient populations, maybe underserved populations, right, that just don't get into the clinic and aren't fully clinically evaluatable in our current paradigm. And so, again, as part of Catalyst, we want to change how we do this. So I'd like to just spend a minute on how we think of the program being executed. So the Catalyst innovators can come from anywhere, right? We're going to have companies that are working on the next generations of in silico AI-enabled ADME-Tox models. We have researchers who are on that cutting edge of understanding patient clinical populations and doing unique clinical and other research activities, building living systems to help us model these things. And then we have product sponsors, and we have the parts of industry in which we want to reach in the Catalyst program. Because while we have a technology challenge in making these tools predictive of human outcomes, we have an adoption challenge, right? And for Catalyst to be successful, the tools that we're building can't just sit on a server or sit on a shelf or be provided, way upstream in drug development. We really want to impact change in those regulated environments. And so to do that, we need an environment that's conducive of adopting those technologies and those tools in preclinical and non-clinical drug development. So these teams are likely to be comprised of multiple members. Could be multiple academic labs in a company, could be 3 companies and a couple academic collaborators. Our business innovation division team is going to talk more about this, but we're envisioning these teams coming together around a membership agreement or a consortia type of contracting approach, which really allows for flexibility in the teams that come in and out. It allows for flexibility about how the work is executed. It's not your traditional prime and sub relationship. And we're doing this because it's very likely that that work's going to have to occur in different types of sprint activities across the first phase. Right. So the first phase of Catalyst, our goal is method qualification, method pre-qualification, getting a capability configured to be able to be used in a good laboratory practice equivalent digital environment. And that's informed by the context of use from a pharmaceutical collaborator, is informed by a capability gap, right? Something that industry needs, right, to really increase the accessibility of these types of tools. And then in the second phase of Catalyst, if your team is selected, really the primacy of that is going to flip to product sponsors. And so ARPA-H in the second phase will not only fund the implementation of those in silico technologies in a regulatory interaction, such as an IND submission, but will pay for the entire IND enabling study package, right? To potentially include doing additional types of data generation to satisfy the review process. And so our end goal at the end of phase 2 is first in human trial approval for a candidate pharmaceutical, right, using the in silico methodologies that we've developed as part of Catalyst. So that's how we see it coming together, and those are the end goals. To do that, we've defined 3 technology areas in which we'd like to focus the thought leadership within this room. So the first technology focus area is really centered around data discovery, data ingestion, and methods to develop better deep learning models to these types of problems. Right, so we know companies are in this space, right, doing drug discovery types of tools. We know there's a lot of data out there that we'd love to have access to that these tools would really benefit from. So TA1 is really an emphasis on how to bring that data together, how to leverage that data for predictive ADME-Tox and PK/PD, and then how do we do this in an environment that's regulated with assurance. Right, so that is the GXP environment that we will be pursuing as part of Catalyst. So good laboratory practice equivalents, right, that exist in the wet lab world. We want to build that environment in the dry lab world. The second technical area is an acknowledgement that while there's a lot of data out there, we probably don't have the best data. We probably don't have all the types of data that we need if we really want to be modeling human physiology. And so TA2 is an investment in living systems tools for model development. So in this technical area, we really want to be focusing on purpose building ex vivo, in vitro clinical cohorts, right, that are going to allow for us to collect the types of data that we really need to get to TA3, which I'll talk about in a second. So TA2 is all about building those living systems methods that are instrumented in a way to collect data that's going to help us simulate human physiology. And TA3 is really where that data and really all of these TAs come together. And that's our in silico human physiology model. That's our, quote, digital twin. Those are physics and simulation-based types of tools that are going to allow for us to apply a candidate small molecule or biologic and understand ADME-Tox and PK/PD within that model. And we see this type of an approach, right, potentially expanding into something like a digital phase 1 trial, for example. Important in all of this is that adoption, right, in those contexts of use and those requirements that I mentioned. And so we are requiring teams that submit against the program to include a product sponsor. That product sponsor is going to help in the first phase of context of use. They're going to help with data generation. They're going to help understand from their pipeline perspective where things might be able to plug in and be a value add. And then in that second phase, right, they'll be part of a team already. They will have had some of those early FDA interactions about how to use these tools in a filing. And then they'll lead the regulatory submissions in that second phase. So we've got 3 separate submission tracks for Catalyst. Option 1 is TA1 alone with a product sponsor. Option 2 is TA2 and 3 paired together, again with a product sponsor. And then option 3 is inclusive of all 3 technical areas with a product sponsor. And so we are running a program called APEX, which is an antigen design program. It was one of our first programs at ARPA-H. And we sort of asked for everything in that program. And what we found was that that was pretty prohibitive into forming teams and going after some of the more discrete challenges within how we would set up something like Catalyst. And so with these 3 options, we've left a little bit of flexibility for teams to come together around where their expertise lies, but really still be supportive of the overall goals of the program. So I wanted to kind of delve a little bit deeper into how we think about executing this program. And so phase 1, we talked, it's a 36-month first phase. We have an independent verification and validation system within this program, right, who's going to be there to provide guidance along the way, but also evaluate the technologies that are being built in those first 36 months. And so we really want active feedback and active participation across the teams. We don't want to get to the end of those 36 months and realize that the tools that we've built are not generalizable, right, are not built around some of the requirements for the program. And so we have an independent arm who's going to be helping us and my team with assessments along the way in those first 36 months. Right, and then the end goal out of that first phase is to get to a qualified novel drug development tool to get to a tool that is qualifiable or placeable within the context of a regulated environment for an IND-enabling study, right? Things that are going to enable for its use in a drug development pipeline. And then teams that are successful, right, in building these tools that are generalizable, that are commercializable, right, that get us to the solutions that we're looking for, it's possible those teams are selected to move to the second phase of the program. And in those 18 months, ARPA-H is supporting regulatory filings for an IND, the IND-enabling studies that go along with that filing, the inclusion of the in silico methodologies, right, and generation of any additional data that reviewers need to see in that second phase to get first-in-human approval using our in silico methodologies. So this is kind of the end state, right? Our end state is to really enable more equitable and patient-focused drug development, right? And we'd like to do that through the creation of tools at this very critical bottleneck in drug development that really prevents smaller market drugs, rare disease therapies, right? Things that don't meet the large market forces that allow for companies to pay millions of dollars and spend years waiting for CRO support for in vivo studies to really access a suite of in silico tools in a GXP environment that are going to be familiar to regulatory authorities. And so our Catalyst funded teams, right? We envision a commercialization strategy that ends in new co-creation or working in a assured GXP environment with contracted services. Those are some of the companies and innovations, right, that we'd like to push into the future. So what I'm going to do right now is dive into 3 technical areas and talk a little bit about what our success metrics look like. I wanted to note something on the last slide. There was a bullet there. Our team is very focused on methodologies that require a higher order mammal in vivo model to demonstrate a surrogate of human efficacy. So those are non-human primate studies. Those are pig studies. Those are studies that include cats and dogs and other types of higher order mammals as a surrogate for human safety. Right, we've demonstrated over time that those tools are not predictive of human outcomes. And so we'd like to focus methodology development for areas that replace those types of models eventually in the future from being used in these preclinical environments. Okay, so let's dive into TA1. So really, there's a lot of data out there. There's a lot of tools out there, right? There's a lot of environments that are very interesting. There's pre-competitive and competitive data that's across all of our, NGO and industry sectors and academic sectors. There's things in publications. There's databases all over the place. And so what we'd like to do for TA1 is really stress the discovery of those data, the inclusion of those data, bringing in novel types of data into the environment, right, that are going to allow for us to train models and build tools that are more predictive. Right, so success criteria for us in this arena looks like discovering novel types of data, potentially novel clinical cohorts, right, things that would really allow for us to be more predictive in the models that we train and build. We'd love for teams to consider metadata standards, synchronization across diverse data sets. I mean, one of the big challenges that we have in this arena, right, is that no one's data looks like anyone else's. Right, so how are we going to manage this in a way that's automated to ingest these data and really learn from what's already been done in the sector? And then we're also pushing, right, for the development with these data as a foundation of novel drug safety models, right, that are focused on these existing data opportunities in an environment that is GLP equivalent digitally, right? So thinking of standardization, assurance, security, right? Things that a GLP environment has that allows the FDA, right, to have confidence in those methodologies. We want to be bringing forward that type of confidence and that assurance from the digital tools that we're developing. So these are eye charts, and I understand the screens are kind of small. These are all in our solicitation. And so when we evaluate abstracts and we evaluate full proposals, right, we are looking for cross-reference against these metrics. Now, these metrics in some cases are pretty aspirational. They're pretty challenging to meet, right? So the reason why we've written them into our solicitation is to really push the proposing teams to think about solutions that are going to get us to the end state that we'd like. And so I'm not going to go through these in a lot of detail here. They're in our solicitation, right? They're spelled out very clearly, and happy to answer any questions about these at our one-on-one sessions throughout the rest of the day. But again, we're looking at, right, repository platform requirements. We're looking for model parameters. We're looking for really the ability to do these things in a GXP environment. So TA2 is focused on enabling technologies for living systems tool development, right? So we're building living systems tools for a variety of different things in biology, right? We're building complex in vitro systems. We're building organs on chips, right? Usually to model disease in humans, to maybe look at drug efficacy, to maybe better understand system communication, right, in the human body. So for the purposes of the Catalyst program in Technical Area 2, we want to build these systems around the data that we need to simulate regular healthy human physiology for the purposes of really simulating a phase 1 clinical trial environment, right? So how do we better understand from our own physiology, right, how our body works? So that could be novel human clinical types of activities. How do we think about leveraging advancements in complex in vitro systems, organs on chips, ex vivo organ systems, even surrogate in vivo models that we have today? How do we better understand how all those things work to simulate human physiology? Right, so this might be a variety of technologies. This might be real novel data collection methodology or instrumentation within some existing workflows. And again, this might be really leveraging clinical cohorts that are instrumented, that have longitudinal sample collection, right, for the purposes of understanding how our physiology around ADME-Tox and PK/PD work and how we can simulate them. So again, we've laid out some pretty stringent metrics here, not to limit creativity, but to really try to push the envelopes of what we know is out there and what we think is necessary, right, to make something like simulating human physiology possible. However, if there are creative approaches that are on the margins of these metrics or something, right, that you have access to or that you're building, which is really unique that maybe we didn't even know existed or could exist, we'd love to hear those approaches as well. And so please let these metrics be a guide for how you think about putting a team together and the technology that you bring in, but it's not exclusionary. So we know we don't know everything that's going on out there. And so we'd love for as much of that creativity to come into the Catalyst program as possible. And so TA3, again, is how do we put all these things together? How do we simulate? How do we have physics-based models built around our human physiology for the purposes of ADME-Tox and PK/PD? So these are generalizable types of models. These are pick a drug, pick a candidate, input into the system, and get a readout of predictive safety and things like PK/PD, which contribute to efficacy. And how do we leverage AI/ML to do this? How do we build better models, right? How do we go from simulating organ systems, right, to whole human types of models, right? There's existing tools that are being built out there at the organ system level, which we think are really cool. We are very excited about figuring out ways to stitch these models together to think about comprehensive whole human ADME-Tox and PK/PD simulators. So again, a lot of metrics, a lot of specifications. Let these be a guide. When we do evaluate proposals, right, we do refer to the metrics. But this is another area, right? One of the exciting things about an ARPA-H program is that we are asking for things that haven't been done before, right? So we know some of the solution space. We've talked to a lot of people, right, working on these problems, but we don't know everything. And so if there's a solution, if there's a tool that really fits the margins of these metrics, please talk to us about it in our one-on-one discussions. Please ask a formal Q&A question. And then, really please think about trying to include the creativity pieces going against these 3 technology focus areas. Okay, so most of that TA work is in the first phase, right? So that's where you're building the tool, you're building the methodology, you're building the platform, right? And you're configuring it in a way that allows for regulated use, essentially, right, in a regulated environment. Phase 2, again, flips to how do we demonstrate these tools work in non-clinical product development, right? So how do we bring a package to a regulatory agency and say, hey, we've included these methodologies. Here's our homework about why we think it's a better methodology than what we have today. And here are the results that demonstrate that our candidate's going to be safe and effective in humans. And so those 18 months are really focused on that. Our success there, again, looks like first in human trial approval based on a foundation of in silico methodologies, right, that we're funding and generating within the Catalyst program. So we also have guidance for our product sponsors, right? So while everyone is interested in the next, GLP-1 drug, those are areas in which maybe equity and accessibility, right, are not at the forefront. So we want our product sponsors that come into this program to have a dedicated, to really have a validated need for these tools, right? So they struggle with getting the populations that they need to model in the clinic how safe and effective their drug might be, right? They are working on a small market, small margin type of therapy where, the $10 million CRO animal study, NHP study is just not accessible, right? They're working in rare disease areas where really understanding clinical populations, right, and really disease and health models are very challenging preclinically. And so those are the types of partnerships that we want to be engendering as part of the Catalyst program, right? So how can we use the tools to really break through these bottlenecks in a way that's going to be effective for getting more therapies out there, safer, faster, less expensive, right, and more representative of human population diversity in preclinical environments. So I mentioned population diversity in different types of human clinical populations. And so, right, typically phase 1 people, 18 to 35, healthy. Phase 2 and phase 3 populations change based on enrollment parameters, right, and the types of populations you'd like to target. But we know we really struggle to include population diversity in clinical studies, right? And we certainly struggle to do it preclinically, especially with animal models. And so we'd like to figure out ways to better represent adolescents, the elderly, pregnant women in preclinical studies, and underserved populations, which we typically struggle to reach, right, doing traditional clinical studies. And so this is just a quick snapshot, right, of how we aim to improve patient-centric drug development. And that's really starting from a foundation of human-centered data and modeling preclinically. Right, so our goal is to really capture that innovation that's happening across these 3 technical areas that's occurring in drug discovery, right? That's occurring in better analysis of clinical trials and enrollment. That's occurring in really complex in vitro and ex vivo systems, right? We want to leverage all of that for the purposes of our comprehensive, generalizable in silico models for predicting ADME-Tox and PK/PD preclinically. So I'm going to keep mentioning equity and accessibility. It's really key for any ARPA-H program, and it's part of our it's how we're measured really as successful programs. And so we have a program underway right now called ACTR, Advancing Clinical Trial Readiness at ARPA-H, which aims to improve on our clinical trial diversity, right? So how do we build these decentralized trials which are able to include populations which typically don't have access to these investigational medications? We want to be addressing that from the opposite side through preclinical study diversity, right? So how do we better represent all the differences that we have, right, in a preclinical environment so that when we get to phase 1, phase 2, and phase 3, and post-license surveillance, right, we're not surprised by different populations having different interactions with the pharmaceuticals that we're developing. And so the end goal, right, of Catalyst is safer drugs, more drugs quicker, and more drugs that are accessible and less expensive because we've cut out a lot of failures that happen along the way. So within this, right, we think that we are going to be constantly improving on the tools we develop, right? And that's one of the reasons why we have those assessment points built into the first phase of our program. We really want to have an active feedback dialogue with teams. We want to be bringing in novel types of data that maybe our interagency partners have access to, right, and provide that to the teams we're working with as part of Catalyst. So we really want to ensure that as the tools are being built, especially that are enabled by artificial intelligence and machine learning, right, we are constantly improving and learning and configuring our platforms to improve over time, even after support from ARPA-H ends, right? Because our success, again, is built on things that are built into products that impact patients, right? Our success is built on the longevity of the success that our investments yield. And so tools that last and are efficacious for a couple of years, right? That's not what we're looking for. We're really looking for ways to change the market forces around these problems and build tools that have long-standing durability and efficacy. So I already covered this, right? We know the challenges today. Our preclinical tools are not predictive of human outcomes, right? And as a result, 75% of clinical studies fail due to safety and efficacy, right? That is a huge cost. It's a huge time. It's an opportunity cost that's lost, and patients are impacted in clinical studies, right? But also for therapies that can't get through that workflow because it takes too long, it's too expensive, and things are not predictive, right? And so we envision a future drug development ecosystem where patients are at the center, even in preclinical studies, right? And we've built an in silico methodology toolkit that allows for us to be more predictive of human physiology and human outcomes to the place where we envision a future where maybe phase 1 trials aren't necessary, right? Or we can look for different endpoints, or we can consider different types of trial designs and study designs based on our preclinical data that we're developing here. Okay, so with that, I'm going to transition over to our business and financial manager, Dominic Brown. He's virtual. He's going to brief through a couple of the minor details around this, and then we're going to dive right into our contracting office, who's going to walk through all the detailed contracting particulars around how we're envisioning catalysts coming together after the source selection process. So take it away, Dominic.
My name is Dominic Brown, the Catalyst Business Financial Manager. So I'll be going over the contracts and the solicitation procedures and the cost proposal, some of the do's and don'ts and what to expect at the cost proposal as you go down through the checklist. So moving on to the next slide, this is kind of the process review that we do for that we're going to be doing for Catalyst. So all your solution summaries and along with your proposals will be submitted through solutions at ARPA-H.gov. And just note here that no solution summaries or proposals will be reviewed prior to the deadline date. Reviews will begin after the deadline date. So both summaries and the proposals, we have compliance and responsiveness review. Pretty much when the review time comes, the team will make sure the format requirements are in place. We got technical requirements and that and main things that is submitted on time. So with the summaries, we will have a subject matter expert that reviews the assignments. And once the reviews are done, PM will make a recommendation after deliberation and make a recommendation. And once a recommendation is set, you will receive a letter that will be encouraged or discouraged for you to submit a full proposal. So we go to proposal sides, pretty much down the list, pretty much 1 through 6 is about the same as the solution summaries, with number 6 being that we'll have an SRO, reviews, recommendation, and determination. And this time, once your proposal will be selected or not selected, and you will also get feedback with both options there of why. And then once you are selected, if you are selected, we will begin negotiations. So move to the next slide. So here's some of the, most of these, program, we're going to try to go through the other transactions for us to contract. So here's some of the required documentation that involves the other transactions. So you had the technical and management document, and we asked that it be about 35 pages. We had the task description document, and of course, there's no limit on that. And so the Gantt chart, we asked that be processed through a Gantt chart we submitted that describes the task and description document. So next one here is the cost proposal. Of course, there's no page limit there. Just make sure you support all your items that's needed in your cost proposal. So the cost proposal spreadsheet, it would be the SL424. Pretty much fill out the tasks that's applicable to you, and then some tasks may not be necessary for you, so don't worry about filling them out. And then, of course, we got the administration necessary policy here, and this is including your human research and your animal research services. And then, of course, you have your model agreement. So next slide, we go into, here's some cost proposal suggestions and reminders, some of the do's. So make sure all your requested information is posted on the cover sheet when you submit your proposal. The cost estimate of your total funds requested for ARPA-H and amount of any cost share. And then this is a big thing here. Please just do the cost breakdown by fiscal year and the phase. So then the next one here is, so any supporting documentation that's above $10,000 that we will need justification. So there is justification, written justification on why this line item is above $10,000. So the next big thing here is just remember that the prime of the proposal and the contract are responsible for the subcontractors' cost proposals, whether that the subcontractor does their own cost proposal and send it to the prime and submit it through the proposal like that, or the prime does the proposal themselves. But the prime is responsible for all subcontractors' cost proposals when submitting the proposal. And then the last one, of course, just include how the proposed costs are consistent with the TDD that we mentioned earlier, and that you have a reflect understanding of the cost and the level of effort that's needed to successfully accomplish the technical approach of the proposal. Next slide. So here's some of don'ts as the cost proposal. So don't change the formulas in the spreadsheet. You change the formulas that could give you your total cost could be messed up. The numbers not adding up in the total column. So no rows, columns in the worksheet spreadsheet. Please do not delete any extra tabs. Please do not delete. If this does occur, the process could take longer for us to review as a team, and even still, we may have to return the proposal back to you and ask for additional information for you to correct that, so which will break the process time even longer. And once again, with these, try not to underestimate the cost of the proposal. Don't try to come in at $10 million when we know realistically it's not $10 million just to try to get the contract. So please keep that in mind. As we move on, here's the cost proposal checklist that we, as a team here at ARPA-H, will also go down and check as the proposals come in as we review. So the solicitation should be included in the cost proposal cover sheet, because we will balance that off that also. And then the travel, make sure all the cost proposal have a major cost breakdown in your cost proposal, your direct labor, your travel, your subcontracts, the indirect costs. And once again, next one, number 6, as I mentioned earlier, the 10,000, make sure we have justification for the 10,000. And then also, if you have any vendor or consultant proposals, include them in the summary builder also, as I mentioned that earlier, along with the statement of work. And any team members consider a federal funded research and development center? If so, does your cost proposal include the documentation demonstrating that, and have the unachievable and that you have exhausted all other options to find the equal capability? So here, this is kind of the kickoff timeline for Catalyst program. As you can see, this is proposals day. So as we move forward, the solution summaries are due November 25th. Once again, as I mentioned earlier, they would not be reviewed on the 24th, or they would not be reviewed on the 25th. They would start reviewing on the 26th. At that time, we would be reviewing all abstracts. We would go through the reviews, make selections where we will encourage or discourage a full proposal. At that point, if you have been selected to be encouraged to a full proposal, the full proposal will be due January 31st. Once again, we will not review anything earlier than that. And after that, of course, we go through the whole process of the proposal review. If your proposal is selected, negotiations will begin. Of course, you'll get a feedback letter saying why you were selected or not selected. And then you are not, I ain't gonna say required, but you can offer if you want to have a feedback session with the team, we can offer that of the feedback. And once again, you can go plus or minus 2 weeks at the end of this while negotiations is being taken place. That concludes my brief. If you have any questions, please reach out and we'll answer accordingly. Thank you.
Great. Thank you, Dominic. Can we go back, Jay, one slide? So I just wanted to leave this up here for everyone to kind of crystallize on. So our abstracts are due on the 25th of November. That's about 4 weeks from today. Our team will be turning those around very quickly, and we will be encouraging and discouraging. We've got about a 2-week window to get those back to you, submissions of full proposals, right? And so after that encourage, discourage comes back to you, you'll have an additional 8 weeks for the team to come together and put a full proposal together. Within those encourage letters, we provide pretty good feedback about some of the things that we're looking for, areas to improve upon, et cetera. And for the discourage, we do the same. We provide very robust feedback in, the areas that maybe the proposal missed or something that didn't just quite fit with the goals of the Catalyst program. We also offer feedback calls as a result. And so we're happy to discuss any of our decisions with everyone after that decision point. So full proposals are due at the end of January. And then source selection starts, right? We have received your full proposals. The ARPA-H process at that point is we convene a panel of government reviewers who will review the proposals against the evaluation criteria. And then we'll come up with proposals that are selectable or not selectable. That process typically takes 2 to 3 months. And after that review is complete, and after we've determined who we'd like to select or not select for the program, we communicate those results, again, with a letter and with feedback, and we provide the opportunity for feedback discussions. After that notice of award goes out, the teams that are selected for the Catalyst program, we start contract negotiations. And those are going to be other transactions. And our bid team's going to talk quite a bit about that. And that is really where, these agreements come together. That's where we align on statement of work details, budget, all of those things that go into finalizing a contract happen after you've been notified that your full proposal has been selected for award. So next steps after today, get the team together, put together your abstract with a rough order of magnitude budget. The cost proposal spreadsheet that Dominic mentioned is a helpful way to do that. We're not requiring that as part of the abstract submission process, but it's a helpful tool to budget around the goals of what we want to do for Catalyst. And then, abstracts are due November 25th. You will get encouraged or discouraged from submitting a full proposal shortly after that. While I'm up here, I'm going to start transitioning to our bid team, but there's a couple of questions. So the abstract submission period is open now. So it's not like you've got to submit it on the 25th of November. If something comes in before then, we'll receive it. We'll acknowledge that, and the review will start after the 25th. And then question about numbers for how many will be encouraged and how many will be funded. Great question. So a lot of that is dependent on what we get in. And so I'm going to give you the classic government answer and say it depends. But it really does depend, because we're asking for a lot here, and it depends on the solutions that are provided and the budgets, right, that are developed by the teams for what it's going to take to do that. So with that, we're going to have a more substantive Q&A here with our contracting office. I'd like to hand it over to Diane Sidebottom, who is our Division Director for the Business Innovation Division at ARPA-H.
Thank you. Good morning, everybody. I'm going to talk a little bit about the contracting process and the kind of vehicles that we're going to be looking for or going to be awarding off of this particular solicitation. And then I want to talk for several minutes about the unique kind of teaming approach that ARPA-H is looking for in this particular solicitation. This one is a little bit different than you may have seen before from ARPA-H or from other government agencies. And so I want to give the opportunity for folks to ask some questions. I'm actually, unfortunately, can't stay all day, but I have Kathy Brodo, who's in the back of the room from my office, who's very experienced in this as well. It's going to be available to answer some questions for you too. All right, so as you know, our solicitations are called ISOs, just in case anyone hasn't seen one of these before. It's an Innovation Solutions Opening. In this particular solicitation, we're going to be laying out all the different aspects of this program. So it's very important for you to read completely the solicitation. We have a tendency to have people that think this is like a traditional program that they've seen in other agencies or that they may have seen from ARPA-H and don't read the whole thing. And we want to make sure that you're as successful as possible with your proposal. So please make sure that you read the entirety of the solicitation so that you know what needs to go into your solution summary as well as your proposal. In this particular situation, we are not going to be awarding cooperative agreements off this ISO. We're just going to be awarding other transactions. Other transactions are a different kind of contracting mechanism. They're not a traditional government contract. And they have a lot more flexibility and a lot more ability for us to negotiate with you about the different aspects of your solution. And so that's the agreement type that we've chosen. If you were to propose and ask for a cooperative agreement, we're going to steer you back towards OTs. So this is not a negotiable point. So just know that going in, and you may want to take a little time to bone up a little bit on OTs if it's unfamiliar to you. And obviously you can always ask questions of us as well. There is no funding limitation for individual OT projects, but within the solicitation, you should understand that obviously there's funding limitations on this particular program, but there is no dictated amount per agreement that everyone would get. The solution summaries, when they say they're mandatory, what that means is that you need to submit a solution summary in order to be considered, have your proposal considered ultimately down the path. It does not mean, though, that solution summaries are going to be like a gate necessarily, where you're going to be told you can submit a proposal or not. As Andy mentioned, you're going to get an encourage or discourage letter with some feedback, and I would highly take that into account when you are considering whether or not you're going to submit a full proposal. If you were to get a discouraged letter, you would probably have to revamp your solution quite a bit to make it successful. And the idea with these encourage, discourage approach is we're trying to help you all. We're trying to make sure that you don't waste time and money on a proposal that you're going to be unsuccessful with. And we want to make sure that you understand exactly what it is we're looking for, or what is it about your particular approach that may not be of interest to us. So take that very much into account. Obviously, we are looking to get solution summaries as well as proposals that are conforming. What we mean by that is it provides all the necessary information that we ask for in the ISO. So that's why I say, please very carefully read through the ISO and make sure that you give us all the information that we've asked for. Otherwise, you may find that your solution summary or your proposal is rejected, and we don't want to do that. We want to make sure that everybody who takes the time and energy to submit something to us gets a chance to be reviewed. Okay, so OTs. I do not have enough time, unfortunately, here to talk about OTs, because I could talk about OTs for days on end. But OTs are a different kind of instrument. And if you are largely familiar with getting financial assistance instruments, for instance, like grants or cooperative agreements, they are quite a bit different. The idea here is that OTs are outside all of the normal acquisition rules and regulations. So that would be outside of the FAR and the agency supplements, as well as 2 CFR 200. That gives it a lot of flexibility in how these agreements are put together and what is included in them. It gives us a lot of negotiation possibility, but also it helps in this particular program because of the more unique teaming arrangement that we're looking for. And so if this is unfamiliar to you, please ask questions. We're more than happy to explain how OTs work. There's also a lot of information on the web about OTs now. I've worked with OTs for many, many years, and I'm always laugh a little bit when people are saying, oh, what is this new thing called OTs? OTs have actually been around for about 50 some years at this point. It's just that not every agency uses them, and not everybody's familiar with them. They are going to look different. They are not going to look like a traditional financial assistance agreement, or even like a traditional FAR-based contract, if you've ever had one of those. So just understand that when you get it, make sure you read through it all completely. We do have a sample OT that we have published on the ARPA-H website that you can go take a look at. Understand it's a sample. That's not necessarily going to look like exactly like the one that you would get, but it will give you a sense of the format and the kind of terms and conditions that are in an instrument like this. So in terms of eligibility, this is an important section to read within the ISO, depending on where you come from. We do have some limitations on certain types of entities in submitting solution summaries and proposals to this particular solicitation. So if you are coming from a government organization, we will not accept a proposal from you. If you feel that you have something to offer, or you feel that you have a solution that would be of interest to us, you need to contact the program team directly. But we will not accept a proposal through the normal proposal channel. That is also the same for FFRDCs, which are federally funded research and development organizations. If you are from one of these entities, again, contact the program team, and they can talk to you about what your participation can look like and how that would work for you. So we're looking to get our proposals from entities that are more commercial-like or academic-like institutions. Everybody familiar with sam.gov? Okay. Yeah, I know. And well, not everybody is usually. So sam.gov is a required government site that you need to register with. It is one of those things that's just unique to the government that I wish I could get rid of, but I can't. But registering with sam.gov is how you're going to get an award and how you're going to get paid, which is usually important to people. And so somebody on your team needs to be registered with sam.gov so that you have at least one entity on your team that can receive payment. And so make sure that that is done. It is not a immediate kind of thing when you go to sam.gov to register. There is a lag associated with the registration. So if you have not registered before, give yourself time. Do it now, because it may take a couple of weeks for it actually to hit sam.gov. And if you are already registered, please go in and check your registration and make sure it's not going to expire before your proposals do, because that's going to be key for us to be able to accept your proposal. So just go in and check. Non-US organizations, we do have no restrictions really on foreign entities from participating on a team. We do have a statutory prohibition on certain countries participating, and that's unique to ARPA-H. Go ahead and look in the solicitation and it will list out those countries. There's only 4 of them. And usually they're not going to be countries that most organizations are going to deal with. But also too, within this particular solicitation, there's an unusual, and I say unusual, not so much unusual for an ARPA, but maybe unusual for many of you, kind of teaming arrangement that we are looking for in this particular situation. So let's talk about teaming. This has been discussed to some degree already by Dominic. I just wanted to highlight it again. Make sure that you take a look at what is asked for within each one of these documents and what the timeline is going to be about these. Make sure too that you answer all of the questions that are asked of you within the solicitation. So that makes sure that we can evaluate you quickly and get you back some feedback. But we're going to be evaluating your submissions based on the evaluation criteria that are listed within the solicitation. And so focus on those. There's 4 of them. So it's not too many. And you'll understand then better how to write your proposal so that all of those questions are answered and we have the right information to look at to do that proper evaluation. Okay, so these are the evaluation criteria that are listed within the solicitation. Overall scientific merit, proposer's capability and relevant experience, potential contribution to the ARPA-H mission, and then a budget risk analysis, which is going to take a look at your costs. This is very similar to what you might see in a broad agency announcement with other government organizations or a notice of a funding opportunity. So this shouldn't be too unfamiliar to you. But those are going to be the 4 things that we're going to be taking a look at when we look at your proposal. Okay, so let's talk a little bit about teaming. So in the traditional government awards don't have a lot of flexibility or a lot of differentiation when it comes to the kind of teaming arrangements that usually are acceptable. But in this particular case, and in ARPA-H in general, we are always looking for different approaches and different ways that we might facilitate teams to provide us with the best possible solutions that'll get us to the goals that we're trying to get to. And in this particular case, Andy thinks it has determined that a different kind of teaming structure is likely going to be more successful for getting to his ultimate goals with this particular program. And so the teaming structure is going to be important. It's going to be important for you to consider how you're going to put this team together. And it's going to take a little bit of time for you to do that. So this is why we wanted to talk about it now. So you have some time to start thinking in your own head about what your teammates are going to look like and how you're going to bring all these folks together. Because it will be something that might be a little bit different than what you're used to. So let's talk about just teaming in general. Next chart. So traditionally within the government contracts that you may have seen before, usually what the teaming structure that we get is a prime sub relationship. So we have a prime contractor who's the one who actually signs the agreement. So they are the party that the government enters into the agreement with. And then they will have sub awardees or sub performers that they deal with themselves and make arrangements with that are not necessarily going to be beholden to the government. It's a fine relationship, and I'm going to talk about the goods and the bads of that in just a second. But in this particular case, that's not really what we're looking for. We're looking more for a multi-party type of arrangement that's going to give us more flexibility as the program progresses to have the right kinds of people leading the effort at the right times. And so this may be relatively unfamiliar to you. For many government organizations, doing something other than a prime sub is very unfamiliar. I have been doing this for a long time. I used to work at DARPA, so we've been doing this for many years, these multi-party relationships, and it can be quite flexible, especially in these kind of collaborative kind of programs like what we're trying to do here. So what's important usually in this kind of relationship, and this is what we're trying to do a little bit differently, is who do we have privity of contract with? I am a lawyer by training, so I'm going to apologize for that right now, and just be honest. But this is really important because this will dictate the kind of relationship that the government can have with the team, because privity is going to be the key aspect of this in terms of who is signing this arrangement. And so we want to make sure that we have the right kinds of folks with the right kinds of right ability for us to reach out and have direct relationships with these people. And that's why privity is going to be important, why we're structuring this a little bit differently. Okay, so prime sub relationship. Everybody should be familiar with this, I would imagine. Yes? You've all seen this before? Okay, so in a prime sub relationship, the government enters into a contract with a single entity. So that would be a university, it could be a nonprofit, it could be a commercial company. And that particular arrangement is governed by federal law, usually, whether that's 2 CFR 200 or the FAR. And then the prime enters into commercial relationships with the individual subcontractors. So the problem with this kind of arrangement, or what makes this a little bit artificial on occasion, is that the government only has that direct relationship with the prime. We do not have any relationship in that kind of scenario with any of the subcontractors or subperformers because they are not signing the arrangement with us. It is a more downstream kind of relationship. And so that can create some artificial constraints. It can create some disadvantages in how programs progress, but it also has advantages too on occasion that the government has utilized over the years. So let's talk about the advantages and disadvantages. So next slide. So why is this advantageous? Well, it's because it's comfortable for most folks. But we don't always like to do the comfortable here. We like to try new things. There is only one person that's responsible to the government in this kind of arrangement, and that would be the prime. So from the government's perspective, in many cases, this is very advantageous to us. We don't have to talk to too many people. We don't have to worry about what a whole bunch of people are doing on the team. We just have to worry about what the prime is doing, and the prime is responsible for making sure that their subcontractors perform in the way that they're supposed to. We only have to negotiate with one person. Hopefully this will never happen at ARPA-H, but only one person can file a protest or file any kind of claim with us. The disadvantages here, and the disadvantages are key for what Andy and ARPA-H are trying to do with this particular program, is it does create a bit of an artificial separation between the government and any of the sub-performers. In many of these kind of programs, the sub-performers are doing a vast amount of the work, maybe some of the most important parts of the work, and yet they're always going to be this sort of game of telephone to some degree of talking to the prime, who then talks to the sub. And the prime can dictate how much interaction the government can have with the sub performers. And that can be a problem on occasion. Also, too, if the prime does not do a very good job, it doesn't matter how the subs are doing, really, it can make this whole thing fall apart. And so that's a little bit problematic. And for this particular program, one thing that the prime sub relationship does not allow is for a change of leadership of the team along the way. There's actually a statute that prevents us from switching out primes. It does not allow us to do that in the government. We would have to terminate the arrangement and issue a new one if we wanted to put a new prime in place, unlike commercial contracts where that's very easily done. In the government, it's just not allowed. But oftentimes in these kinds of arrangements, particularly in a program where the life cycle is trying to get us from the research stage all the way to the actual implementation stage, the same people that are important and who could lead this effort at the research stage are not necessarily going to be the right people to take it to those next levels of the life cycle of the technology development. And so this kind of teaming arrangement, the prime and sub arrangement, does just not allow for that switch in leadership as the program progresses. And that's going to be important in this program because that is key to what's listed in the solicitation is what the acquisition strategy is. So what we're looking more for in this particular arrangement, next slide, is a multi-party teaming arrangement. And so with this type of arrangement, the government does not enter into the contract with any single entity. It enters into the contract with the team as a whole, which means that every member of the team has privity of contract with the government because all of the teammates are signing the contract. Now, from a logistical perspective, that doesn't happen. It's not like I'm going to send the agreement around to every single member of the team and ask for them to sign it. What usually is going to happen in this type of arrangement is that as a team, you will come together and you will decide who's going to be the lead member. And that's totally up to you. And that lead member is going to change as the program progresses, probably. But that lead member is going to be the one that acts as your agent. So in other words, you empower them to speak on behalf of the team with the government. Trust me, this is much easier than trying to have a team of 20 people trying to talk to the government all at the same time and trying to have everybody sign. And that way, that lead member can sign on behalf of the team. Generally, that lead member would receive payment on behalf of the team and then would parse out the payment to the various members. And then in that way, we have that direct ability to interact with all of the teammates, but all of the teammates are also responsible for the solution. In a prime sub relationship, just the prime is. But in this relationship, all of the teammates are going to be equally responsible for the success of the program. There's a lot of advantages of this type of relationship. And quite honestly, this is a more common and understandable relationship for a lot of folks from academia, I've discovered over the years, because there's a lot of collaboration that happens within the scientific community, within academia. So this is not necessarily going to be as unusual, but in this particular case, we're going to have teammates that are obviously going to come from commercial industry as well. And so this will be something that you will need to sit down and talk with your teammates about how you're going to structure this team and what that team arrangement is going to look like. Because it is really important before we can sign an arrangement with the government and the team, that the team has an arrangement amongst themselves. You're going to need to have some kind of legally binding arrangement between you all that will say what your responsibilities are, how that relationship is going to be managed, and all of those things. Many of the things that you're going to have to negotiate with the government, you're going to have to negotiate amongst yourselves first. So coming up with a teaming arrangement, oftentimes it's called an articles of collaboration. You can call it whatever you feel like. I don't really care. That is going to need to be laid out and resolved before we can sign the arrangement with the team. It is also important that you pick a lead. In the early days, when I used to do this with OTs back in the 90s, sometimes people would want to just have sort of this like socialist kind of environment where no one was actually in charge, but everybody just worked together. Yeah, it doesn't work so well. You need to have somebody who's kind of in charge and making sure the trains are running on time and everybody's moving in a common direction. Now, who that leader is totally up to you. Who that leader might become as the program progresses, again, is up to you. To some degree, the solicitation has some expectations at certain stages within the program that whom the leader might be. But as a general rule, the government is not going to get involved in your teaming relationship or your teaming agreement. It's not something the government's a party to. We're going to leave that to you all to decide how you want to handle that. And so make sure, part of why we were doing this here today is because we wanted to give you all as much notice as possible about this more unusual type of arrangement, because you all are going to need to spend some time putting your teams together. Now, does your team have to be fully formed in its end state the day that the agreement is signed? Absolutely not. But you're going to need to have your core members at least, and you're going to need to have a relationship established. Do you have to be a legal entity? Do you have to create like a partnership or a corporation or something in order to sign with the government? No, you don't. You just are going to need to have some kind of contractual arrangement that binds you all together towards this common goal of working on this program with the government. Can you have people join the team later? If you'd like, sure, absolutely. Can you have people drop off the team later if their part is done or they just decide they don't want to play anymore? Sure. That's all possible. This is all largely going to be dictated by what you all think is going to be the best solution for your particular approach and for your particular teammates. So there's a lot of flexibility with these teaming arrangements. There are some examples out on the web. We're going to post an articles of collaboration example just so you can see it. I'll be honest with you, it's an old one that I have. I don't particularly like it all that much, but at least it gives people an idea of what they look like and where they might start from. But there's a lot of examples on how to do this, and you shouldn't feel constrained necessarily on how you choose to approach how you put your team together. But what is going to be important is that you all come together as a team. You establish this relationship, because we're not going to be able to sign the agreement with you until that agreement is signed by everybody that's going to be participating at that moment. And so also within that relationship, you're going to want to establish when the leads can shift, because within this program at a certain point, the lead member is going to shift to the product sponsor. I have that right, Andy? So, okay. I read a lot of solicitations, so I want to make sure. And so how that's going to be handled and how that's going to be established is something that you're going to want to think about. Now, you don't have to have your product sponsor when you propose. That is something that you have a little bit of time to discover or to make that or suss out that relationship. But you're going to need to have within your teaming agreement some mechanism by which this is going to be accomplished. One thing that I will be cautioning you about, this is not something the government is going to help you with. This is not something the government's going to write for you. I can't tell you how many times people have asked me to just write it for them and then give it to them. No, don't want to do that. Don't think that's right, actually, because everybody's going to have their own approach and are going to have different needs. Please don't give the government a job in this agreement. I've seen many teaming arrangements where the government is like your disputes authority. I'm not going to do that for you. You're going to have to figure that out amongst yourselves. Because we're not going to be signing this. We're also not going to be dictating any of the terms, and we're not going to be making any edits or reviewing it. This is your relationship. You just need to establish that. What I will ask, though, is that I'm going to need to see the signed cover page because I need to know that it was executed before we can sign the agreement with you. And that should be the extent of why we need to see it. The only other reason why we might ask to see it is just to make sure that you didn't give us a job somewhere in there that we didn't sign up for. But other than that, it will be your relationship. And so you should think broadly about this and what would be the right approach for your particular team and for your particular solution and how you might best effectuate that. So don't think narrowly on this. There are not a lot of constraints on how this can be put together. And we are certainly not going to dictate too much to you, but just understand that you're going to need to have, at least on your team, one person that's going to have a cage code, that's going to be registered in SAM, that's going to be able to receive payment, and is going to be able to take on that leadership role and monitor the team throughout the program until it's time to switch leaderships to the product sponsors. Okay, so obviously everything that's said here today, whether it was said previously in the last presentation or in mine, is going to be subject to whatever's in the final version of the ISO. So if there's any conflicts, which there shouldn't be, make sure that you read the ISO, and the ISO is going to guide. If we make any amendments to that, we will publish them in SAM.gov, and we'll make sure that everybody has access to them. Hopefully, we'll resolve any issues that we have by having this Proposers Day, and we won't have to have too many amendments. If you have any questions, you're going to have the ability to ask those in just a second. I'm on time, so you should have plenty of time to ask. And then, as I said, Kathy Brodo is going to be here all day, and she's perfectly more than capable of answering any questions that you might have about the teaming approach, about OTs in general. But if you have any questions after this, make sure you use this email address, and we will get back to you as quickly as we can. And then again, I can't emphasize enough, just read the ISO altogether. Please don't assume that it's similar to what any other agency is doing, because there's some unique aspects to this program. Okay, so I was told to leave enough time for people to ask questions, so I'm going to open the floor. You ask any questions that you like about teaming, about OTs, anything like that, please feel free. Yes, ma'am.
So for the product sponsors, since these are typically pharma companies, right? Do you allow for them to submit a full budget for their part of the work? Are they required to do any cost sharing?
So cost sharing is always appreciated in submissions. There's not a requirement in this proposal for cost sharing. It's very possible, right, that as part of this teaming agreement, there might be some data generated or some things that in theory would cost the product sponsor something that might not be something that we would fund necessarily through the agreement. But yes, that's okay, but it's not a requirement in the program for a product sponsor to have cost sharing.
So for the initial budget, we can let the pharma or the potential pharma partner just make their budget and have it reviewed by you guys so you can see what is eligible or not, or do we give them any? I didn't find any instructions for the pharma budget as a product sponsor.
Yeah, so the consortia as a whole should have a budget. The team should have a budget, right? And that should include all of the participants. Now, we are giving a little bit of flex in timing for formally signing up a pharma partner. And so when you read the ISO, you'll see you have essentially 12 months to bring in a product sponsor partner if they're not resident at the beginning of the program. After that 12 months, if they're not part of the team, then the agreement gets terminated. And so we definitely encourage for those collaborators to be there from the beginning, especially as their input will help strengthen an abstract and certainly a full proposal. But those budgets should be built into the team's budget that they submit through the full proposal process. And so most of what Diane mentioned is very critical at the full proposal and contract negotiation stage. For the abstract, we don't expect the team to have to come together. We don't need to see that collaboration agreement or anything like that.
For the product sponsor, you mentioned already, I just confirm do we need to include that even in the abstract submission stage, have to identify who's the product sponsor, number one. Number two is, when we submit, do we need to specify which drug product specifically, or just general or partner?
So it helps at the abstract phase, right? If you have a partner identified who's in some way, agreed to talk about the problem. We also don't require at the abstract phase like letters of commitment or support or anything like that. And your second question was related to
a specific product rather than just a partner.
Yeah, so we do have some requirements for a product we might want to evaluate in that second phase, and that's in the metrics section of the product sponsor part of the ISO. We don't need a specific like molecular structure or identifier or anything, but a general concept of the product pipeline that would be evaluated as part of the second phase would be helpful to have.
Thank you.
Yeah, so relevant to previous questions, if you're thinking about involving a pharma, then the intellectual property, right, can be a really sensitive point. So just wonder whether, you claim any IP or you require any disclosure of any of the information here, or how's the requirement here?
Yeah, so it's a great question. So for the first phase, that method development part, we understand, right, that for these technologies to actually impact on how we do things, they need to be commercializable. You need to be able to sell that capability right through sort of a CRO model into the sector. We are requiring that the raw data that gets developed that will be used to build those tools will be open sourceable, will be shareable. But then as we get into the second phase, any product candidate that's evaluated with the tools, we only ask that the results of those assessments be published. So we're not asking for any of the underlying intellectual property or anything that a product sponsor might bring in terms of sharing that more openly with the community.
One thing I will mention, though, and one of the reasons why we're using OTs is we have a lot more flexibility in negotiating intellectual property. But having said that, we do still have certain key requirements that we're going to need, and Andy's just described them. And that part of the agreement is oftentimes the longest part of the agreement when it comes to negotiation. I wouldn't worry so much about it, the details of it in the solution summary stage, because there we're just looking for a summary of your approach. But when you go to do your full proposal, we're going to ask you for some underlying like background data restrictions for you to lay out. So that will be something that you're going to want to start to think about so that you're prepared for your proposal.
Hi, thank you both for your presentations, or all three of you. Two questions, mostly around, again, product sponsors. The first is that we all know that drug development can be a little, unreliable on timeline. So first is like, is it permissible and is it encouraged to have more than one product sponsor or a product sponsor with more than one asset in the pipeline to sort of mitigate that risk? And second question is, in the proposal, it looks like the intention of these things is to replace IND-enabling tox and ultimately like phase one ideally. But is that the intention in the first turn of the crank, or will they be doing IND-enabling tox studies in parallel?
Yeah, so great question. On the first point, 100% encouraged to have multiple product sponsors with multiple requirements, multiple contexts of use, right? Our end goal is generalizable tools. And I think, the teams that are coming together would like to be building those as well. And so having multiple product sponsors, having, a pipeline that potentially includes, some NGA or academic candidates that are looking to get into first in human. So yes on that. And then the second part of the question, yes. So the question was, do we intend to replace with the first shot on goal essentially in the second phase, or do we anticipate doing things in parallel? We've left that a little bit open. The goal of the program is to replace tools that are not predictive. Now, we understand, right, working with regulatory agencies, across the world, that has different flavors in terms of replacing those tools. And so it's likely to be somewhere in the middle. If we build enough of a data package in the first phase, and we have a tool that becomes qualifiable, potentially replacement is there. But ARPA-H will support parallel studies or studies that come after reviewer comments in that second phase.
Hi, Bill from Medical College for Wisconsin. So this is the question for Andy, and it looks like the program put a more emphasis on safety study over efficacy study. Is this a correct impression? And may you elaborate the rationale behind this? Thank you.
Yeah, so you're correct that 1A is safety, especially with the title of the program, including ADME and Tox. We also have some PK/PD aims, and all of those things contribute to efficacy in some flavor, and so that's one of the reasons why efficacy is in the program. The primary endpoints, though, are safety. One of the reasons why we moved away from efficacy is that gets us into disease modeling, that gets us into discovery, that gets us into other types of model development that were out of scope for what we were thinking, and so you're going to actually hear from some of our government collaborators who are building tools to go after some of those questions. And we thought that was an area that industry is moving into, maybe doesn't need the government emphasis per se. And so, yes, our primary endpoints are safety, although we do think with the comprehensive tools that we'd like to build as part of Catalyst, efficacy becomes part and parcel of that.
I have a couple of questions. One is about the scope. Basically, as standards, we have animal testing that's still being done. Is there a scope on this program to conduct some parallel animal models that compares the industry standards with what we do in vitro?
Yeah, so it's a great question. So just to repeat, the question was, will we allow for in vivo experimentation, right, to demonstrate equivalency or anything like that? It's likely to be required to some degree. We don't really know to what degree. And so, one of the challenges in this program that we're punting to you all to solve is, how do you build these tools that are not just equivalent, but also superior? And how do you demonstrate that? And what kind of data goes along with that? I think a lot of that might be direct regulator team interactions around a workflow to get that done. But yes, we would support additional in vivo experimentation if necessary.
And my second question is about international collaboration. I saw from the presentation that you can have collaboration outside the US as well. Is there any legal complications on that in terms of signing these agreements?
No, not really. There is some considerations that we will have to take into account. Like I said, there's a statutory requirement for us. There's certain countries that we cannot make awards to. And those are listed in the solicitation off the top of my head. Honestly, I can't remember what they are. But there is also some reviews that we will have to do to make sure that the level of influence from certain prohibited sources isn't occurring. And so every proposal that we get is going to have that to some degree. But in terms of normal participation by a foreign source, no, we do have a bit of a domestic preference within our statute that our director is required to consider, but that doesn't necessarily mean that that's going to limit any foreign participation or that's going to necessarily knock anybody out of consideration. So I wouldn't worry about that so much unless you have any particular limitations that you're concerned about.
No, and I think especially for companies operating in different regulatory requirements around the world, some of these contexts of use or capabilities that we'd like to build around here might be more applicable to EMA statute or what the EMA is looking to do. And that's great. I've worked with both the FDA and EMA and other international regulators, and so would love to consider proposals, right, that think about how to get these tools in a way that's acceptable across the world. However, we would stress that FDA remains, a part of the plan for sure.
Right, and to some degree, we are spending U.S. taxpayer dollars, so we want a lot of the value of the results of this to flow back to the United States, but that doesn't mean it can't flow globally as well. So it just can keep that in the back of your mind.
Over here. Yeah, thank you very much. Great presentation, super clear, very exciting. Some of the questions I have were already asked, but I want to clarify a little bit on team structure and on IP consideration. So exciting to hear that it's encouraged to have multiple drug sponsors come, but how does that work? Because if there's perhaps some firewalling that needs to happen there, that's okay and understood. Not that not every team member would necessarily be working as long as the lead is coordinating that well. And then is there flexibility once you get into phase 2 that those might, like individual drug sponsors then sort of take the lead for moving that forward. Is that an accurate description of how that might work?
Yeah, that's the intention, actually.
Right.
To the second question,
Yeah.
that the product sponsors would take the lead.
Right. Okay.
Now, if you have more than one, logistically, you're gonna have to figure out a plan on how that's gonna work. But I don't think that's necessarily something that you have a preference on, is it?
No.
Okay.
And that's one of the reasons for this type of teaming approach, right?
Right.
Is that, again, we'd like to stress generalizability of tools that we're developing. We know that priorities change, right? So if we require everyone to have a prime sub relationship with a product sponsor, upon submission, in 2 to 3 years at the end of phase 1, companies go out of business, strategic directions
Right.
change, right? That would cause our relationship between the government and the prime to be really challenging.
Right.
And so we want all of those approaches to flexibility around IP, what gets shared, what doesn't. I think all of those are open for consideration.
Okay. So the point is maximum flexibility and effectiveness. That's fantastic. One last quick question on that. So in terms of IP considerations between team members, is that a part of what we should work out on our own before
Absolutely.
it comes to you guys? Yeah.
Absolutely.
Excellent.
But with the understanding that he has certain requirements
Right.
for his program that the team as a whole will have to be able to accomplish. But as amongst yourselves, how you share amongst yourselves, what kind of relationship you want to have about that, that's really left up to you all. Now, we do discourage people doing like forced sharing just to become part of a team.
Yeah.
Because that's not really the kind of relationship that we're looking to foster. And remember too, your team doesn't necessarily have to have all teammates be at the same tier necessarily. I have seen teams before where there is a core group of members that stay with the program the whole way through. And then there's other members who may have a lesser position, still part of the team, but may come and go, or may have not as many voting rights or whatever
Got it.
that might be. So all of that's up to you all on how you want to do that.
Great. Thank you very much.
Yep.
We have a few questions online. First, is the expectation that the administrative lead may change over the course of the program, or does that position remain stable while the technical lead shifts?
Administrative lead can
Yeah,
change.
Absolutely, the administrative lead can change. Some of it is dependent too on how many teammates you get. I've seen teams before where there becomes enough teammates that it's difficult for any one performer, and you want that person to be a performer to handle the administrative aspects of it. So it may require that at that point lead shift to somebody else. But yeah, we're not limiting it to administrative or technical. It's all one package.
And related, is it a requirement that the lead members switch between phase 1 and phase 2?
That's not a requirement. It's a likely scenario, which is one of the reasons why we're pursuing this type of teaming arrangement. But no, it's possible that one person has a solution for everything, which would be interesting to consider.
Another question, if the team lead is responsible for distributing funds to members of the team, is the team lead also responsible for the financial reporting and overseeing team member expenditures?
Not necessarily. There can be a split in responsibilities in that regard, but generally that is the case, that the lead team member is also going to be responsible for any financial reporting that you have, just because they are the ones who are handling all the money. But I don't think that's necessarily a requirement. If you have a good reason why that needs to be split up and it looks like it's going to satisfy our needs, I don't know that we'll care all that much.
Are there limits on the size of the team overall?
Nope.
So yeah, no limits on size of team. There's a world in which there's a number that's unwieldy and difficult to manage. So no, we want teams that are, collective groups that are pursuing the same goal together. And that could be 3 teammates, and that could be 10 teammates. And really the ability to have people that come in and out during the course of the program is one of the things that we want to stress here too.
So I can tell you, I've done a lot of these kind of teams, and we've had teams as large as like 300, 350 members. At that point, it starts to get a little unwieldy, and it's a lot to manage. So I'm just throwing that out there, but we're certainly not going to limit how many people that you have.
Andy, you also mentioned you will cover the cost for IND enabling from the product sponsor. That's include GLP-Tox, GMP manufacturer. That's number one. Number two is if you have a multiple partner, that cost could add up quickly.
Yes, so that's something teams are going to have to consider because we want the best opportunity to demonstrate that these tools are going to translate. IND enabling studies is what's within scope within that second phase, and so not the full set of things you need to do to satisfy an IND submission requirement, right? So like the manufacturing, the GMP manufacturing, that would occur in a separate bin that we wouldn't sponsor, but we would sponsor the IND enabling studies in which things like the in silico methodologies we're pursuing would potentially replace or augment the tools that are used today.
But not in GLP-Tox.
No, yeah, GLP-Tox would be part of that IND enabling study packages.
Thank you, Andy. I think my question is there are two questions. One is, what's the role of the product sponsor in the phase 1 stage? And the second is, in the budget, do we need to include everything up to stage 2, like in the, let's say, abstract submission? Because it's really hard to foresee what's going to happen later in the stage, right? And INDs, and these are like big budget.
It's a great question. And thank you, Diane. She's got to run. But we've got Catherine and any other bid-related, contract-related questions we'll be able to answer. It's a great question. So in terms of the budget, we are asking for teams to be thinking about both phases at the abstract submission. The way that we've configured the effort is that we would like to be able to roll directly into phase 2 upon completion of the objectives and upon success. And so we don't want to have to put out a separate solicitation. We don't want to have to recompete. And we don't typically do like unpriced optionality in our contract agreements. And so considering at least with an abstract, it's an estimate. It's a rough order of magnitude estimate on budget. Considering what it would cost to get phase 2 completed is critical. And then your first question?
What's the role of the product sponsor in phase 1?
So the product sponsor in phase 1, I think less of a like technical doing the work type of a role, and much more of a, this is what we need. Like, this is what we need the thing to do. This is what we need it to look like. This is what's going to satisfy our regulatory affairs part of the organization. These are the things that we would love to adopt the tool that's going to make our processes better. So there's advisement there. There could be some data generation. They will also likely lead for the team interactions with the regulator, right? So if you have like a pre-IND discussion and you lay out your set of experiments that you want to do, obviously you want to include these types of methodologies within there and get advice from the FDA, for example, about what they might require in terms of data to demonstrate that tool can be part of an IND enabling study.
Great talk, Andy. Very informative presentation. I have a question regarding the drug development tools. Before the sponsor to endorse that, how we handle the conversation? FDA had to go through the quantification program to fully say you can use that as a way in the R&D package. So how we lay out the arrangements in the proposal in terms of that?
Yeah, so it's a great question. So the FDA has multiple mechanisms to get a novel drug development tool reviewed and potentially qualified. I would say that that's not the only mechanism with which a tool might enter into a place that's more qualifiable for like an IND submission. And in fact, people use many tools that aren't necessarily qualified, right, to demonstrate safety in IND enabling studies. So I think it kind of depends on the focus of the tool, something like the commercialization strategy for how we want this tool to be used in the future. There's a few different things to consider. And so while entry of a tool into a review pipeline in phase 1 is great, right? That might not necessarily be a requirement or something that a reviewer is going to need to see in the second phase. And so that's an option for the teams, and we would certainly support that. But that's not the only route for these tools to be adopted in the second phase.
I see. Can I have another one?
Yes.
So regarding sharing all those data, sharing all those platforms to the public at the end of the program. So if there is a IP related issue for the sponsors, is there any wiggling room for this delay sharing if the sponsors have hesitation to disclose everything during the process of developing the products?
100%, right? So like Diane mentioned, we're using the OT mechanism that is a lot more flexible than some of the traditional government contracting types of vehicles that we use. And so things like IP, especially if there's intellectual property, design data, et cetera, things that are within some of the data sets that we would want released, we can work around some of those parameters. Yes.
Yeah, just a quick question about whether you anticipate any cross-program sharing of information. I think we will be embarking into the unknown with respect to validation of in silico modeling. I mean, Part 11 compliance in current GLP settings is a challenge in itself with emerging technologies. And this could be a great opportunity to synergize with agency objectives, and wanted to know, what you might anticipate happening.
100%, right? So we absolutely want to create an environment where our selected teams are working basically together, because they're, while we hope one team is able to solve a lot of these problems, right, it's very unlikely. And in fact, one of the reasons this is an ARPA-H challenge is none of these proposals that we might select might work, right? This is a very type of a high risk arena. And so we would want those teams to work together. We're executing other programs across ARPA-H in which we're doing this, and we've built the structures to allow for the data to come in a common environment that allows the team to learn from what's going on across the program. So 100%. The last question I'll take online, question regarding scope. Should the models focus on a point where the lead or candidate is already in hand, or would scope cover models to forecast target mechanism-based toxicity and guide drug design? So I think that's a question that I think we would leave open to the teams to determine. If you think the best opportunity of success of the tool that you're building to fit in a regulated digital environment and inform drug development, right, is for something that you already have, that you've done preclinical experiments on, but you haven't done IND-enabling experiments, possible. If it's, there's a target that you can drug and you really want to do that, and you want to use these tools to help guide that process, but then also guide the regulated environment space, I think that would be okay too. And so I think we kind of are, the way that the program is configured, we're more aligned towards the non-clinical routes. So things that are maybe past the discovery phase, but I wouldn't want to discount any applications that could span both those parameters. Okay, so with that being said, it's been 2 hours of a lot of information coming at you, and so really appreciate the questions. Any other Q&A questions that we don't get to cover, please put them in the chat. We are recording all those, and we will answer those in our Q&A page. Take a few minutes, and then we'll reconvene here at 11. Okay, everyone, let's come back into the main room, please. So thank you, everyone. We're getting our virtual panelists configured right now. But we are very excited to have not only the ARPA-H team here today and working on the goals of Catalyst, but we also have colleagues from across the U.S. government who are interested in leveraging these emerging tools to do better with animal experimentation sort of across the biomedical R&D apparatus. And so, I'm really excited to have their participation in the Catalyst program. And what I'd like to do is to give them a few minutes each to walk through some of the other efforts and other coordinating activities that are happening across the U.S. government. So the first speaker is going to be Nicole from NIHS. She's one of the co-chairs of the ICFAM, and I'm going to let her discuss in depth what that is and how their activities in the 3 Rs related to animal experimentation relates to the goals of Catalyst, and other opportunities for the group here in this room and the group virtually to engage with what they do, to learn from a lot of the great work that's already been happening across the ecosystem. And there's a lot of really good resources in animal use and R&D experimentation and goals in the 3 Rs across that space, and those resources are available for all of us. So Nicole's going to join us here shortly. I'd also like to introduce Margaret, who's a program officer within the Common Fund office at NIH. She is running the complementary program, which is a really unique approach to transforming how we think about in silico and chemico and in vitro types of systems when it comes to really innovating on non-in vivo approaches to research and development.
Great. So my name is Margaret Ochochinska, and I want to thank Andy and the ARPA-H team for the opportunity to present at the Proposers Day today. So I will tell you about a program that NIH has been developing that's in the related space to Catalyst. And so this is the Complement Animal Research and Experimentation Program. And I am part of the Office of Strategic Coordination at NIH and the Office of the Director, and it's in the Reengineering the Research Enterprise Division. So very appropriate for what we're attempting to do. So I wanted to tell you a little bit more about the Office of Strategic Coordination, or the Common Fund. So initially it was launched as the NIH Roadmap in 2004, and Congress unanimously reauthorized the NIH in 2006 and established a Division of Program Coordination, Planning, and Strategic Initiatives within the Office of Director, and the NIH Common Fund to provide a dedicated source of funding to enable trans NIH research. So while before institutes had to contribute funds for joint programs, this created a dedicated source of set-aside funds for trans NIH research. And so the mission of the Common Fund is to move the NIH mission forward faster, supporting bold scientific programs that catalyze discovery across all biomedical and behavioral research, advance areas of biomedical and behavioral research important to the missions of multiple NIH institutes and centers, and to spur subsequent biomedical advances that otherwise would not be possible without an initial strategic investment. And so the features of the Common Fund programs to catalyze biomedical discovery are to accelerate emerging science, remove research roadblocks, enhance the research workforce, and support high-risk, high-reward science. And so the way we do this is through collaborative management processes. So scientists from diverse disciplines provide input as we plan the new programs. Researchers participate in interdisciplinary consortia to tackle shared goals. And their leadership and staff from institute centers and the Office of the Director work together to design funding opportunities and oversee the projects in a joint fashion. And so the purpose of complementary is to catalyze the development, standardization, validation, and use of human-based new approach methodologies, NAMs, that we're all familiar with, that will transform the way we do basic translational and clinical sciences. The goals of this program, as Andy alluded to, is to better model and understand human health and disease outcomes across diverse populations, develop NAMs that provide insight into specific biological processes or disease states, validate mature NAMs to support regulatory use and standardization, and to complement traditional models and make biomedical research more efficient and effective. And so this was brought to you and made possible by a rather large working group that spans over 23 institutes and centers and offices at NIH. So you can see a very broad representation of working group members. And this activity was led by the two co-chairs, Joni Rutter, the NCATS director, and Rick Wojcik, NIEHS director. And I'm the program leader for this concept. And also working group coordinators included Dan Tagley and Dan Shaughnessy. And we have a strategic visionary that will be speaking after me, Nicole Kleinsteuer, who was also on the working group. And so again, this was a large trans NIH effort, and I will talk a little bit about the timeline. So essentially, just in brief, we had multiple activities on the left-hand side for strategic planning that included deep landscape analysis, listening sessions, roundtables across to hear from multiple stakeholders across the private and public sector. We also held an interagency workshop and had an ideation design prize. And then ultimately that resulted in concept approval earlier this year. This was in parallel to the NIH Advisory Committee to the Director Working Group on catalyzing the development and use of new approach methodologies to advance biomedical research. And they also held workshops and convened meetings to develop recommendations that were approved by the NIH Director in February of this year. And so essentially the program goals span multiple areas. So we are interested in vitro models emulating population diversity, in silico multiscale systems, and chemical cell-free systems, as well as combinatorial NAMs and integrated fair data sets and AI engines. So the scope is very broad across all of these areas. And so this kind of builds on new approach methodology activities across NIH. So NIH has funded multiple activities across all these areas, including digital twin models, in silico models, complex in vitro systems, and in chemical screening. And these were all brought together to create an integrated solution. So this is what complementary is aiming to do, is to integrate all of those different areas into one potential platform for further development. And so, as I mentioned before, we had multiple strategic planning activities, including a listening session that included academia, industry, NGOs, government, international partners. And then we had this interagency retreat that included NIH, FDA, EPA, NSF, and ARPA-H, of course, and BARDA, VA, DARPA, NIST, NASA, CPSC, and ICVAM that Nicole will talk about. more later after my presentation. And we identified some scientific needs, priority areas, including chronicity, neuroscience, personalized health, cross-disease pathogenesis, and population diversity, as well as operational needs needs to integrate, coordinate, and collaborate, which includes shared data infrastructure, standardized frameworks, validation, and training. The other planning activities, which you can see here, and there are also links there to the Ideation Challenge Prize that we held, and we have winners that were announced as part of this prize earlier this year. And we also did a landscape analysis where you can access the summary report at this link. And so for the challenge prize, we conducted this earlier this year. We provided a total prize purse of $1 million, with each winning team receiving $50,000 for their innovative solution. So this was an ideation prize for potential solutions that could be used to inform our strategic planning process and development of the program goals. And so you can see more information if you use the QR code or follow the links in the presentation. And so in brief, based on all of the strategic planning efforts across NIH, we developed a program with multiple components. One is the Technology Development Center to stimulate the development of NAMs to fill areas of greatest need, with emphasis on increased biological complexity and throughput, innovative combinatorial approaches, and data sharing. And data sharing is very important across this program. So we also have a data hub and coordinating center to create integrated data structures, including standards for modern career to improve fairness of NAM relevant data, and to create a searchable NAMs repository. We also have a third pillar of the program that is a validation and qualification network, or VQN, for NAMs adoption and implementation. It's to establish common data elements and standardize reporting, to apply validation qualification frameworks, and to accelerate development and deployment of implementation of NAMs in the regulatory space. And also community engagement and training will be very important throughout all of these pieces to promote the development of an inclusive, diverse biomedical research workforce with the skills to build and use NAMs, community engagement, and societal and ethical considerations. And so this kind of shows a visual representation of the NAMS Data Hub, serving as the central role to coordinate the various centers. And I'll tell you briefly about some funding opportunities that are coming up around this. So we'll have a funding opportunity for the Data Hub, as well as technology development centers. And the technology development centers will be comprehensive, including pilot projects, integrated NAMS technology development, community engagement, and the various cores within the centers. And we are also working with the Foundation for NIH to establish the validation and qualification network for NAMS adoption implementation, to include various agency partners to coordinate the broader landscape in the NAMS space. And so the first opportunity I want to tell you about is the Technology Development Center Notice of Intent to Publish. So you can see the link here as well as the QR code. So estimated release date is around Halloween or thereabouts. So please be on the lookout for that. There's the various links here, and you can also sign up for the listserv to get notifications about this. The next slide, please, is the NAMS Data Hub and Coordinating Center. So there's another notice of intent to publish this funding opportunity around the end of November. So along the same lines, feel free to check out this funding opportunity, as well as sign up for the listserv if you're interested to learn more about the complementary program. And so the last piece is the NIH-FNIH public-private partnership for the validation qualification network. It's really to develop this public-private partnership to bring industry, NGOs, nonprofits into the network by leveraging existing FNIH infrastructure to enable pre-competitive data sharing and potentially supporting validation activities across labs and locations for specific use cases for implementation, to support community outreach and training, and to provide a fluid funnel for potential solutions which may be developed through the comprehensive centers or elsewhere, that other entities that could become part of the validation and qualification network or the complementary consortium. We are interested in including additional federal partners and to synergize and coordinate with other global activities in NAMS. And so this is going to be done through a three-phased approach, starting with the design phase in this calendar year, FY25, to plan, develop the network with industry, NGOs, CROs, and federal partners, to determine the scope of the validation efforts, develop governance criteria, to refine the goals, and to include the ICFAM support subgroup to advise on validation steps and also hold workshops with stakeholders later this year. This is all going to lead into the implementation phase 1. So it will be informed by the design phase to implement the validation efforts with partners to select use cases across all of the NAMS categories, to fund the use cases, and to also interact with the data hub and the training components across the consortium, and to develop long-term sustainability plans. And so that leads us into implementation phase 2, which is a continuation of the initial phase, to apply reporting standards. Very important to think about standardization and ways to apply that across NAMS categories. Also, investigators to interact with the regulators, industry CROs, to nominate mature NAMS for validation, and to support additional training opportunities across the partner groups. And so really, this is just a summary of the flow. So the purpose of complementary is to provide a fluid conduit from stakeholder engagement to implementation, including the 3 pillars: technology development, centralized data resources, and validation and qualification, with training and dissemination being a very important substrate across the path. And I think this is not well presented here, but essentially the purpose of complementary will be to innovate, integrate, coordinate, and transform. So those are the basic hallmarks of what we're trying to do. We want to work with our federal partners across the stakeholder community. So it's a great opportunity to present this at this Proposers Day for your consideration. And a next slide just shows you ways to contact us, to visit the website, and to reach out to us with any questions. So that's the end of my presentation, and I'm happy to take questions if there's time. Otherwise, feel free to reach out at the email link provided.
Great. Thank you so much, Margaret. So these slides are being recorded. We'll disseminate the slides. And so, yeah, the links and the email addresses will be accessible for everyone after the meeting. Great. Thank you so much, Margaret. I believe we've got Nicole on the line. So Nicole, I would love to turn it over to you. We're very excited to hear about ICFAM. Thank you.
Absolutely. Thanks, Andy. And my name is Nicole Kleinstra. I'm the director of NICEDM and of ICFAM. I'll explain to you what those are. And I'm really happy to be talking to you about sort of the existing efforts in new approach methodologies. I realize NAMS is not a familiar acronym for some of you, that we're doing across our federal family. And I want to talk about some examples of where we've had some success, and then some resources that I hope will be useful for folks who are considering applying to the Catalyst program. So this is what I like to call the NAMS confidence continuum. I think it's really important to really have dedicated investment and resources along every aspect of this continuum. Margaret just gave you a really nice overview of complementary, which is one of the programs that is, supporting the development of these new approach methods, or NAMS, in the biomedical research space. My organizations tend to work a little more along that latter half of the continuum to try and validate and qualify new approach methods to ensure their implementation into regulatory decision making, both domestically in the US, but also internationally via organizations such as the OECD. So we are the U.S. validation body. So we were established by Congress in 2000 by the ICFAM Authorization Act. We're made up of 18 different federal agencies that either require or consider chemical safety data and are interested in moving away from an exclusive reliance on more traditional animal models that are lower throughput and perhaps not as relevant to human health, and moving towards adopting more new approach methods. So in vitro, in chemico, in silico, and combinations of those that are going to provide more rapid, more efficient, and more human biology-based information on chemical safety. ICFAM is this committee of different federal agencies. Oh, I see that what Margaret also observed, the formatting is a little off on a lot of these slides. So hopefully most of the information will still come across. So ICFAM is this congressionally mandated committee, 18 different federal agencies that were listed on the last slide, pretty much all of your regulatory agencies like FDA, EPA, and then others like DOD, NIOSH, OSHA, CPSC, lots of different institutes and centers that are part of NIH. ARPA-H isn't part of ICFAM yet, but maybe they'll join. We get agencies joining quite frequently. NICEDM is sort of the workhorse of ICFAM. So ICFAM doesn't necessarily have a budget But NICEDM is a national center within the NIH that provides operational and scientific support to ICFAM, and we have a budget of about $5 million a year, and you can see the group pictured down there. There are multiple different ICFAM work groups, so this is how ICFAM is basically organized. So we have our principal agency representatives nominate subject matter experts to be on these different work groups. So we have work groups around particular endpoints like acute toxicity, or eco-tox. We have work groups around specific chemical classes, so perfluorinated substances, obviously a very high concern. And so looking at developing and identifying new approach methods that are suitable for testing things like PFAS. We have work groups that are formed around strategies and analysis approaches. So how to go from in vitro data and extrapolate that to an in vivo scenario and try and interpret that information in more of a public health context. One of our work groups, the validation work group, has been very busy over the last 2 years publishing this new guidance document. So on the left-hand side, you see our strategic roadmap that we published in 2018, really intended to be an evergreen document. It's pretty short. It's only like 15 pages. But on the right-hand side, you see our new guidance document on validation, qualification, and regulatory acceptance of NAMs. This is a much more extensive document that really goes into the nuts and bolts of how to validate or qualify new methods for particular regulatory contexts of use. And we had, all 18 federal agencies weighing in on this. This work group was led and co-chaired by the FDA, the EPA, and CPSC, and NIST. So it really represents kind of best practices and consensus among federal agencies, particularly regulatory authorities, in how to go about establishing confidence in new approach methods for their implementation into regulatory policy decisions. One way that we've tried to operationalize the concepts in the validation guidance document is through this activity called the Method Developers Forum. And so this is a way to encourage communication between developers of new tools and new methods, be they computational, be they experimental, and the intended end users of those methods, both in regulatory decision-making and in regulated industry. So we are doing this in sort of a topic-specific way. So we started with cancer. We started with looking at methods that could help assess the carcinogenicity of, drugs or environmental chemicals or food contaminants, et cetera. And the first thing that we did was we actually asked our agency partners to summarize their needs around carcinogenicity and how they currently make decisions. So what is their current framework and what kind of data do they use? And where are there opportunities to improve upon that process and opportunities for NAMs to provide, better information, more rapid information, et cetera. And then we asked method developers to view those pre-recorded presentations and then develop their own presentations around their methods with considerations around how they could fit into those various contexts of use that were articulated by our agency partners and our industry partners. And that was recorded. It's all available on the NICEDM website. It happened in August of this year, but that was just the first of a series. And the next topic will be on cardiovascular modeling and cardiovascular toxicity. Then we'll move through to developmental, neuro, specific target organ, et cetera. If you go to the next slide, I think that's just the slide about kind of the process. So as I mentioned, we have these pre-recorded presentations. They're available on the website. That in and of itself, I thought was a really nice sort of deliverable or product to come out of this, to have agencies really talk about like what their needs are and what their decision processes are. And then all of the method developer presentations were also recorded and are also available on the website. So those may, represent potential partners for folks who may apply for complementary funding or catalyst funding. So this is just an overview of kind of all of the portfolio of work that NICEDM and ICFAM are engaged in. I'm obviously not going to talk to you about all of these things today, but I just wanted to pop it up here because if you have, a particular interest or you're developing models or methods in one of these areas and you would like to talk to us about some of the resources that we have available and the research that we're doing, I'd be more than happy to talk to you. And I would encourage all of you to subscribe to our newsletter because it's a really good source of information about sort of publications in the field, but also funding opportunities, webinars, et cetera. One other thing I wanted to highlight is, one of the things that we've been really focused on over the last couple of years is this opportunity for human biology-based NAMs to better address heterogeneity and variability and susceptibility across diverse populations. So we actually have a special issue of in the journal Human Genomics, where we've already started publishing papers as part of this special issue around NAMs that actually can better model population variability. So I wanted to pop that up in case folks are interested in that. So I'm going to talk through a couple of examples of kind of how we've used this collective kind of crowdsourcing approach to form these global collaborative projects to build computational models for toxicological endpoints. Because my understanding of Catalyst is that that's kind of one of the approaches that's being encouraged. So building these teams to build, take an ensemble or a consensus modeling approach to tackle hard problems and in the space of, drug development. And so here we did that for tox endpoints. So we looked at endpoints like endocrine disruption, so binding to the estrogen receptor and causing estrogenicity or anti-androgenicity or acute systemic toxicity. And so we've done this over the course of several years where we develop a very rigorous, very high quality training data set, and then we release it to all, globally, to over 35 different groups around the world that specialize in computational chemistry. And then they use their preferred feature set and algorithm to build quantitative structure activity relationship models to predict this particular endpoint. And then we give them sort of the chemical identifiers for an external test set. They give us the predictions. We bring all those models back in-house. We evaluate them qualitatively and quantitatively. And then we build an ensemble model that leverages the strengths and compensates for the weaknesses of any one individual modeling approach. And this has been tremendously successful, both in terms of generating very robust models that provide prediction for any chemical with a defined structure in the chemical universe, but also in terms of implementation into regulatory policy. So there's this EPA's endocrine disruptor screening program white paper where they talk about using these globally crowdsourced models, SERAP and Compara, as lines of evidence for determining endocrine disruption of substances. In the EU under REACH, they prefer actually those model predictions as kind of the first line of demonstrating the mechanism of toxicity of a potential endocrine disruptor. And they're looking at accepting the collaborative models for acute systemic toxicity in place of the rodent test. So if you go to the next slide, to dig in a little bit deeper to that most recent project, which is CATMOS, we basically took the variability in the training data. So we had over 1,000 compounds where we had repeat animal studies. And so we were able to use a data-driven approach to deriving a 95% confidence interval around those data points, and then use that in evaluating the model predictions. So, the take home there is that those globally crowdsourced consensus models actually perform just as well as the repeatability of the animal studies, but also to put 95% confidence intervals around the outputs of that predictive model. We then work with regulatory agencies. We're working with the EPA to evaluate the utility of those predicted models in their risk assessment applications. So this is a Bishop et al. Paper that just came out a couple months ago. And basically the take home here was that EPA has determined that these consensus QSAR models, if they predict that a chemical is non-toxic, that's a very, very reliable prediction. And we're expecting this to form the basis of a waiver guidance that they're going to be putting out, hopefully in the next couple of months, saying that if you have, a chemical that's predicted to be non-toxic by this consensus QSAR model, then you can waive the animal study. All of these models that I've talked about are available in the Open QSAR app. So here you can see the toxicity endpoints that I've already talked about, but then there's also tons of other endpoints like physicochemical properties, properties that affect absorption, distribution, metabolism, and excretion, environmental fate parameters. This is a suite of open QSAR models that's available through the NIEHS GitHub. And these are very robust models trained on large data sets that have been highly curated. And so again, I think that ideally these will maybe be resources for some of you, particularly when you're looking at things like intrinsic clearance and plasma protein binding that really affect pharmacokinetics and toxicokinetics of drugs when you're trying to model their properties in a preclinical space in a computational way. So you can access ARPA in a bunch of different ways. You can access it through the NIEHS GitHub, through the FDA's Precision platform, through the EPA's CompTox Chemicals dashboard, and through the OECD QSAR toolbox. The other way you can access it is through the NICEDM's integrated chemical environment. And this is kind of a dashboard by which you can access our data, but also lots of workflows. So I'll show you in a minute some screenshots of it, but just so you can see what type of data are available in ICE. We have physicochemical properties and ADME parameters. This is the experimental data. So we have the predictions for all of these endpoints, but these are sort of the data sets that those predictive models were trained on. We have legacy in vivo animal data for cancer, for developmental and repro tox, for various topical toxicity endpoints. But then we also have in vitro high throughput screening data where the assay targets have actually been mapped to things like key characteristics of carcinogens or hallmarks of cancer or mechanisms of developmental toxicity. We have a highly curated version of the high throughput screening data that incorporates things like the analytical chemistry, QC information, but also that mechanistic target mapping to modes of action and biological pathways. And then, as I mentioned previously, we also have these predictive models from ARPA. And so if you don't want to download ARPA as a standalone software and run it yourself, you can also access ARPA predictions through the ICE dashboard. So we've gone ahead and pre-run ARPA on over 1 million substances. And then we also have exposure predictions. So human exposure predictions that are derived from NHANES biomonitoring data and then machine learning models that are trained on biomonitoring data and also on things like material safety data sheets from large retailers like Walmart and use cases. Here's some screenshots of what you can do in ICE. So you can explore high throughput screening data. You can overlay curves on top of each other based on chemicals or target groupings. You can look for a chemical similarity. If you have a data poor substance and you want to look for similar data rich substances, you can run virtual animal models, virtual animal studies, or virtual human studies. So you can run physiologically based pharmacokinetic models and ask, like, what would the concentrations in various tissues be after exposure to a molecule, be it a drug or be it a chemical. And you can do that in a steady state way, looking at steady state concentrations in various tissues, or you can do it in a time course way. So looking at Cmax and area under the curve. And then we also have this consumer products, chemicals and products database on our backend where you can look for the presence of chemicals in various consumer products and goods, and then overlay those and compare them with things like bioactivity signatures from high throughput screening data. Margaret already mentioned the advisory committee to the NIH director NAMS working group. And this was their charge, basically, is to kind of look at the landscape of NAMs and then make some recommendations to catalyze their development and use. These were their recommendations. And so it's really, really heartening to see how many programs are kind of popping up that are, I think, really targeted towards implementing a lot of these recommendations. So improving our ability to protect public health and better predict human disease pathways and come up with new therapeutics via the use of NAMs. One of their major recommendations was around providing access to NAMs, so having this infrastructure that will allow people to both identify and access NAMs for their uses. And so one of the new programs that we're launching here at NICEDM is being supported by NIEHS by an award from the NIEHS director, is to develop a new interactive database of NAMs that have been validated or qualified for various regulatory contexts of use. And this is going to be called CAMERA, the Collection of Alternative Methods for Regulatory Application. So my hope is that, projects that get funded under complementary and under Catalyst that, demonstrate great promise and then become validated or qualified for regulatory application will find a home in CAMERA, and that will be supported by NICEDM and ICVAM. I'm going to stop there and just say, I have this amazing group that I'm really privileged to lead. And if you're interested in kind of exploring some of these resources that I've talked about today, subscribe to our newsletter, go play around with ICE. I hope the data and the workflows and the models in there are useful to all of you as you're thinking about kind of developing your own models. And then if you're interested in kind of a high level overview of all of the things that the various federal agencies are doing in the space of NAMs, you can read the biennial report. It's actually available as an interactive website. So you can go and you can search by like metadata keywords. So all the articles in there are tagged by agency and by topic area. So if you're curious in what, FDA is doing in the space of, development and reproduction or in stem cells or whatever, you can look for that. Okay, I will stop there and hopefully we have a little bit of time for questions.
Nicole, thank you so much. Really appreciate the presentation and a fantastic set of resources, I think, for all of our teams. I've got what looks like one question here.
Thanks. Well, thank you very much. It's a really impressive talk. Really enjoyed a lot. So my question is, it seems you have done a lot on this toxicity prediction already. And my question would be, suppose, I'm a farmer, new farmer, I have this drug X, right, a chemical. And they use all these nice tools to predict it's safe, right? So it seems it's okay for the EPA already that I can weigh my animal study, it's safe in the environment. And now I'm trying to take in front of FDA for the first in human study. So what are the gaps then I still need to cover, and what other evidence I still need to demonstrate for the FDA, before I can put it into the first in human study?
Yeah, I mean, it's a great question. I would say that FDA is going to be your best resource for that, right? So one of the things that we talk about quite a lot in the strategic roadmap and in the guidance document on validation and qualification is early engagement with the regulatory decision makers that you're targeting. So reaching out to them and saying, hey, I'm going to be using these models and these methods to predict safety or to predict efficacy for my new drug candidate. Can we talk about whether or not that's going to be acceptable to you, and can we work together to kind of develop a plan? And so that would be my major recommendation. I think that particularly in the drug development space for some of the newer therapeutic modalities like biologics, et cetera, where you don't have those targets in animals, there is a really great opportunity for using human-based models, both computational and complex in vitro models, to demonstrate efficacy. Safety is a little bit of a higher bar at this stage. So we have models for like topical toxicity and acute systemic toxicity and endocrine disruption, but we still don't have, models that are accepted by regulatory agencies for things like toxicity to embryonic or fetal development, or, the potential for causing cancer, or for like systemic, chronic systemic toxicity. So there's still a ton of work to be done in the area of developing more predictive models for sort of more complex toxicity endpoints.
Great. And agree 100%, Nicole. So thank you very much for the overview. I really appreciate it. And those are all some fantastic resources for all of us as we pursue the goals of Catalyst. And we'll make sure that those slides are disseminated so people have all of those links. And so finally, for this session, I'd like to invite Raj from the FDA up. He is the director of the Quantitative Medicine Center of Excellence. So Raj, please take it away.
Thank you very much, Andy, and you all for providing an opportunity for us to come and share with you our newest initiative, that is the Cedar Quantitative Medicine Center of Excellence, which was announced in March of this year. So before I go ahead, I will try to level set here as to what we mean by quantitative medicine. We define quantitative medicine as a development and application of exposure-based biological and quantitative modeling and simulation approaches, which are derived from a wide array of data sources ranging from all the way from non-clinical, clinical to real-world sources of information, with an intention to inform the drug development, regulatory decision-making, and patient care. And we believe when appropriately applied, quantitative medicine approaches can help streamline and accelerate drug development throughout the product lifecycle. Most importantly, help with identifying, quantifying, and addressing uncertainties earlier in the life cycle and eventually contribute to the totality of understanding of drugs' benefit risks. So quantitative medicine at Cedar has had a long history. Cedar has been at the forefront of advancing many of these approaches to inform the pre-market product reviews, our post-market product assessments, as well as developing and implementing policy. What I will try to provide here is, at a very high level, some of the efforts, recent efforts in the Cedar in advancing quantitative medicine. The first I want to bring attention is to the Model-Informed Drug Development, or MIDD initiatives, which have been very actively used as part of informing drug development decisions, as well as informing regulatory decisions or various elements of regulatory decisions. We have a standalone program called as MIDD Paid Meeting Program, which provides an opportunity for sponsors to engage with the agency in applying various quantitative approaches for addressing issues such as dose selection optimization, or using clinical trial simulations for informing trial designs, or mechanistic safety predictions. From our office, and this is led out by the Office of Clinical Pharmacology and the Office of Translational Sciences. From our Office of Biostatistics, we have a program called as Complex Innovative Trial Designs, and this is a program that allows sponsors to explore and evaluate various novel trial designs, understand their performance characteristics, as well as use them in instances where conventional approaches may not be feasible. That's another program which uses a lot of quantitative approaches. Together with the Office of Biostatistics and the Office of Translational Sciences, we have a fit-for-purpose initiative, which is essentially aimed at developing these dynamic drug development tools as part of drug development, so as to be able to use these tools to help inform the trial designs, such as informing patient selection. For example, we have one that has been deemed fit for purpose in the Alzheimer's disease space for informing trial designs, several methods for informing how to conduct a dose response analysis, and various approaches for deriving dose response information. Many of these that I have talked, the three that I have talked so far are in the pre-market space. We also have a lot of emerging application of quantitative approaches in the post-marketing and the life cycle space also. And within our Office of Generic Drugs, there is a pilot program which is called as Model Integrated Evidence Pilot, which applies quantitative approaches to be able to help evaluate bioequivalence assessments, especially in situations where conventional bioequivalence studies may not be feasible. All in all, what I'm trying to present here is that we have an established history of applying quantitative medicine approaches across various different offices in Cedar. And what this provides us with an excellent opportunity to coordinate and bring the synergies together to maximize their application within the center. And this was the motivation for us to stand up the Quantitative Medicine Center of Excellence, which is a Cedar-wide enterprise to facilitate the continuous evolution and consistent application of quantitative methods for drug development and regulatory decision. And the hope is that by having a consistent application and continuous evolution, we would be able to spur innovation, advance medical product development, inform regulatory decision making, and eventually promote public health. What this initiative does is it brings together 5 different super offices within the Center for Drug Evaluation and Research to be able to coordinate the activities in this space. These include the Office of Biostatistics and Office of Clinical Pharmacology and the Office of Translational Sciences, the Office of New Drugs, who are the major recipients of some of the outputs that come from the application of these quantitative medicine approaches, and those include the Office of New Drug Policy as well as the clinical offices. From the Office of Pharmaceutical Quality, we have the Offices of Quality Research and Quality Assessments, and we are seeing increasingly the use of physiologic-based biopharmaceutics modeling to help inform the product specification and characteristics. And lastly, from the Office of Generic Drugs, I touched upon the Model Integrated Evidence Pilot, the Office of Research Standards, which does a lot of these quantitative approaches. So the technical focus of our efforts include various quantitative approaches ranging from the conventional pharmacometric analysis, such as population pharmacokinetics, exposure response analysis, you name it, mechanistic modeling approaches such as quantitative systems pharmacology, as well as applying machine learning and artificial intelligence approaches, and also the clinical application of conducting clinical trial simulations and using in silico predictions. One of the core focus application areas for us also is to elucidate the biomarker endpoint relationships, which have been very critical in the rare disease development areas also. The current focus of our efforts can be organized in these 3 main buckets. I'll go through each and every one of this in a bit, starting with applied science policy. The focus here is to lead the development and implementation of evidence-based scientific guidances and policies and develop best practices that will foster consistent application of quantitative medicine approaches. To go along with it, our goal is to build a robust knowledge management systems that catalogs the precedents, which will be complementary to the best practices and standards that would be developed, as well as disseminate this information that would allow for stakeholders to envision and identify opportunities for application of quantitative medicine approaches. The second key area is multidisciplinary education and exchange, and these include educational enrichment activities, not only internal but also externally, to be able to raise awareness, to increase the uptake, as well as promote the implementation of quantitative medicine approaches, as well as engage with the community, such as the current forum at large, to be able to learn about the novel cutting-edge approaches, as well as share experiences and incorporate these emerging approaches to support the continuous evolution goal of the Center of Excellence. And lastly, to strategically plan and coordinate various activities in-house as well as with our partners, especially in identifying priority areas for the research, as well as to be able to pool the resources to coordinate and elevate the existing activities. So I hope this, in a nutshell, provides a flavor for what this new Quantitative Medicine Center of Excellence is, and who are all the key participants in this new initiative and the focus of the efforts. I wanted to briefly touch upon what we heard today, And clearly, I think there is an opportunity for collaboration here. Heard from Andy earlier, the vision and focus for Catalyst. And if we were to compare and contrast with the scope and focus of Cedar Quantitative Medicine Center of Excellence, there is a healthy overlap. I think the goal here is to left shift the efforts and utilize quantitative approaches to be able to efficiently inform drug development and decisions. So clearly there is a tremendous opportunity for collaboration. That is my last slide, and I'm happy to take any questions. If you want to know more about Cedar Quantitative Medicine Center of Excellence, just Google it, and you're going to find the website and the information associated with it. Happy to take any questions.
Great, Raj. Thank you so much. Really appreciate the overview and completely agree that, as FDA modernizes and as the goals of the Quantitative Medicine Center are there, I think there's a lot of collaborative opportunities with the groups that we're going to be supporting as part of Catalyst. We've got a couple questions in the room, if that's okay.
Sure.
Thank you for your talk. Really interesting. I am curious, what would be the best way to interface with you or other folks at the agency to get these questions through? Because I know typically industry interactions are highly regulated, and you don't want to, step on any toes or break any rules, but there might be some ad hoc questions that would come about outside of, type A, type B, et cetera, meetings.
Sure. And what I have been remiss in my presentation, I realize, is I have not provided specific contact information. What I would suggest is if you Google for Cedar Quantitative Medicine Center of Excellence, it'll take you to our landing page, and you have our contact or the inbox email address also. And I would think, feel free to just shoot an email. Look, if it is a topic which is highly regulated, confidential in nature, and we are not able to engage, we will let you know. If these are broader questions in general, which are to better the scientific understanding, or in general, try to identify where there might be resources that we could point to, we would be very happy to point out those. So I would just encourage emailing the inbox for your information, and hopefully we can help you in directing you to the right resource, or provide you with information, or connect with the appropriate people in the agency.
Great, thank you, Raj. I think we've got time for maybe one more question.
Thank you, Roger. This is Dushin Song from University of Michigan. Nice information. How we can approach you, for example, if somebody doing machine learning model, they need data sets. FDA has all the data sets, but nobody else can access. So for this proposal, also for the question for Andy, do you encourage us to work with FDA to have access some of the data sets who can do the machine learning, or those discouraged because FDA cannot receive funding?
Yeah, so Raj, maybe I'll take a shot at that one first. So we're working directly with the FDA, right? And we're working out what that looks like as part of the Catalyst program. And so there might be opportunities, right, for program performers to learn from some of the information that's out there. But Raj, I know that, right, probably some of the goals of your office are to make those tools more accessible to industry and academia. So I'd love your thoughts on how you guys are doing that.
Yeah, so I'm not sure to what extent I would be able to directly be addressing the question, but what I wanted to touch upon are those 4 programs that I talked about. There are standalone programs. If there are activities associated on a specific drug development, then we have a pathway called as Model Informed Drug Development Paired Meeting Program, and you can come through that. If similarly, the Complex Innovative Trial Designs Program. If there are questions about methodologies, tools that are being developed, we do have a specific program for that, which is the Fit for Purpose initiative, wherein we provide a forum for engagement and help determine if they are fit for the purpose of application and wherein they can be used, such that when they are applied, we will not have re-litigation of what are the technical aspects and things of that nature. So there are standalone programs for different objectives, not only within the Office of Clinical Pharmacology Biostatistics, but across the Cedars. So I would encourage people just to ask, and we can probably more often than not identify the right entry point and the right program for engaging in conversations.
Great. Okay, Raj, thank you so much. We really appreciate it. Let's give a round of applause for our colleagues.
Thank you.
Okay, so that concludes the morning sessions. Really appreciate everyone's attention and the discussion that we've had so far. If you go to the Catalyst webpage on the ARPA-H website, there's about 100 teaming profiles about the companies, the academic groups, the NGOs who are working in this space, contact information right in the areas that they're interested in. So please think over lunch about, what are the really burning questions that you'd like to ask me and the team about, and we're excited to have those opportunities to talk. With that, again, really appreciate you all being here. This is a fantastic morning session. Really excited to hear some of the thoughts and things that are happening out there in the ecosystem, and we can't wait to see your abstracts in a few weeks.